you will be joined by Prakriti and Vaiba, is that right?
Yeah, it's Prakriti and Vaibhav, yes.
Perfect. So as soon, okay, so I already see Vaiba, so I will make him a host, a panelist.
Yeah.
Can I also briefly share my screen?
Yeah, sure.
Okay. Does it work?
Yeah, we see it.
Yeah, it works.
Okay, perfect. How do I leave this sharing mode?
You should be able to see stop sharing. But
yeah, stop sharing, yeah. Yes, okay.
Okay, and then here is Prakriti.
Yeah.
So for whoever joined in the meantime, good morning. We had some trouble starting, but now we are in time and we are ready to start. So let's do it. The first talk is from some participants that were at Neuromatch Academy this summer, and they are Rishika, Prakriti, and Vaiba. And they will give an interesting overview of several methods to decode activity from neural spikes and more. So there are three of them and they will do the presentation together. So who starts?
Hello, am I audible?
Yeah.
Yeah. So, hello guys. So should I start?
Sure.
Yeah. So hello guys. This project was done as a part of Neuromatch Academy and was done by the four of us. However, one of us couldn't make it due to some work commitments. His name is Narottam Singh and he's affiliated to the JSS Academy of Higher Education and Research in Mysuru. Yeah. So coming to the project, first we come to the question of why motor coordination is so interesting. The answer to this involves its enormously complex nature. Motor control and coordination is a fundamental characteristic of all animals, but it is still a field of active research. This is due to its intricate and complex nature, which involves directed coordination between a large number of muscles and consequently a large number of brain regions. Another reason why it's so complex is the fact that motor control is made possible by highly overlapping coordination and sometimes redundant coordination between multiple brain regions, with some regions contributing more to the motor output than the other regions. So coming to the specific question we wanted to ask, the question is that can we predict the motor output by studying a number of neurons from different brain regions, and also if the information represented by the different regions of the brain is equal or not. So coming to where we obtained our information from, the dataset we used for our analysis was the Steinmetz dataset, which was published last year in Nature. The dataset is from a left-right decision-making task where the mouse had to execute a motor task by moving a wheel placed in front of it towards the left or right based on visual stimulus in front of it. The neural spikes were recorded from a large number of brain regions using multiple neuropixel electrodes. The trial consisted of two parts, one with correlation between the optic flow and wheel motion, and the other one with no such correlation. We focused on the open loop period where there was no correlation in order to avoid the stimulus-related artifacts. I would now like to call Rishika Mohanta to take over.
Sorry, Rishika, you are muted.
Hi, am I audible now? Okay. Let's take a deeper look at the dataset itself. We know that there are a large number of brain regions that are involved in motor control, including but not limited to the cortex, thalamus, basal ganglia, substantia nigra, and the cerebellum. The Steinmetz datasets hold a large number of recordings from multiple brain regions. First, we limited ourselves to regions with at least 50 simultaneous recorded neurons, and through a survey of literature, we identified regions that have been reported to be involved in motor control. Other than cerebellar recordings, we were able to identify multiple subregions and from other important centers in the dataset, although with different number of neural recordings and sessions and mice, as shown as a figure on the right at the bottom. We also observed observed that there was a bias in the data set where a majority of the duration of the trials, there was no motor output, that is a stationary condition, but the number of left versus right outputs were balanced. Now, since there is no one best way to answer any scientific question, we chose to take a pluralistic multi-pronged approach. We hypothesized by taking two different pipelines, namely a spiking-based approach, which we call the population spike code, and a latent population coding approach, which we call latent information code, we would be able to regress to the same outcome. Both processes include a random sampling of neurons and trials from different brain regions in order to ensure the comparability of results once we have them. Now, the first approach that we took was a population spike code approach. The aim was to look at the individual spiking neuron activity of different neurons in a population, which we sample, and see if these spikes together can predict the motor output at the next time point. Now, this involved building a general linear model that learns the temporal kernels that convolve the spiking activity and sum up together to give the motor output. This to give the motor at the end of each window of activity. The first thing to note is that the length of these optimal window kernels is something that can vary from different region to region. And it's not something that we can assume to be uniform across all regions. So we had to first check if that was true. So what we did was sweep through different window sizes, different kernel sizes across different regions, and used cross-validation to evaluate the how good the prediction accuracy was, and found what the optimal window size was for different brain regions. These results are summarized in the figure on the right to the top, where we find that different brain regions have different lengths of optimal history. And this is highly variable across sampling, but the means are slightly different across different brain regions. The other thing that we observed after we saw this was that spikes from different brain areas are only able to roughly predict the motor output, but to different degrees. Although the predictions are not perfectly accurate, as you can see from the R-square scores on the right bottom, we can say that there are differences in the ability of different brain regions to be able to predict the motor output. For example, if you look at the primary motor cortex and secondary motor cortex, MOP and MOS respectively, and you compare them to other regions like MD, mediodorsal nucleus, you find that they have very different abilities. While one fails, the other does a much better job. The other thing that we could infer from this data set is what the nature of the information stored in the spiking itself was. How did we do this? So for this exact reason, we looked at and performed analysis of the learned temporal kernel and find that the amount of information encoded by the spikes is different across different brain regions. How we quantify this is by looking at the variation, that is the standard deviation is what we calculated, and the distribution of the kernels of the sampled population, and quantify the mean variation and the entropy across the duration of the entire temporal window. We found that regions such as the VPM consistently show higher diversity of kernels than many others in both the entropy and the kernel variation approach. The entropy we used was Shannon entropy. We also tried another parallel metric, which was the PCA participation fraction, in which we looked at the number of principal components that were required to explain 90% of the variability in the temporal kernels. Now I will leave it up to Prakriti Nayak to tell us about the latent information code approach, which is our second approach.
For our second approach, thank you, Rishika, we wanted to extract later dimensions from our population spiking data to build trajectories over time, model as a Gaussian process. So we implemented a pipeline to compute later dimensions of the spike data from around 50 randomly sampled neurons from around hundreds randomly sampled trials using GPFA, which is Gaussian process factor analysis, and LDA, linear discriminant analysis, to classify directional output in the lower dimensional space. That directional output, as you can see, could be right, left, or stationary. Can you please change the slide? Yes. So this figure shows the measurement for optimal dimensionality for GPFA for 5 randomly sampled regions, and it's fitted with sigmoid. You can see there's a lot of variability across regions, while the GPFA fit saturates at different rates for different brain regions, but around 20 dimensions seem to be enough. So that is what we are going to be following throughout the project from now on. Here, you can see that motor output has been mapped on GPFA representation. We have segregated into best representation and worst. You can see we have pointed out the same regions, but those are different samples. So some regions separate motion better. So you can see it within the best ones, within the worst ones, they can separate motion better. But at some places, the samples separate better than others. You can see on the top, the primary motor cortex, and the bottom as well, another sample of primary motor cortex, but they are showing vastly different separation. So in order to account for the imbalance in the data, can you please change the slide? Yeah. In order to account for the imbalance in the data, we used balanced accuracy and weighted F1 to see classification is accurate and specific. Some regions have a much better accuracy for motor output than others. From the whole project, it was from Rishika's part, it was unconcluded that the spikes from different brain areas roughly predict the motor output to different degrees and encode different amount of information. These differences could be further explored by looking at patterns in temporal kernels across different regions. And through the latent information code part, we can infer that these regions do not separate the output equally. And using a larger sample and perhaps larger trial size could result in a better classification accuracy and help tackle the regional discrepancies in the results. Lastly, while our results represent movement output, we cannot infer explicit representation of the covariates that you generally see in motor papers, such as the target position that you normally see in the Steinmetz task, and the endpoint, and the mouse joint kinematics, and the muscle activities. All those covariates that's normal in a motor function paper. We can't see that because that's a function of single neuron recordings, and we are doing a population study here. So in that note, we can actually say that any much further exploration can be done with the data set, and as well as these pipelines, we can use this to answer more nuanced questions. These are our references. We would like to express our gratitude to entire Neuromatch team, to Conrad, to Brad, to Dan, to everyone else for making sure that anime was a reality. A special thanks to Jason, who actually guided us through multiple steps that we went through, and we had 5 days to complete this project throughout Neuromatch Academy, and our TA Shagun, who taught us everything basically. And these amazing cartoons, the colorful cartoons that you saw throughout the presentation, those were by Zcard, and we edited those a little bit. You can find his account in Instagram. Our code is available on GitHub, and our poster you can find it here on Figshare. You can follow us on Twitter and LinkedIn. So we started from Neuromatch Academy, ended at Neuromatch Conference. Thank you.
Thank you very much for the talk. It's a very interesting project, and it's very interesting that you managed to do this out of Neuromatch Academy. And you're now presenting it at the Neuromatch conference. So let's go for questions. I have a question. So for the different performances in separating motor output based on the latent neural modes, do you have any interpretation aside the chance of maybe using a larger sample? Do you think that there may be another reason for these differences?
That's a very good question. Now, if we were to look at the results that we see, there are many different things that could be different other than just sample sizes, because the variation in classification ability that we see might be depending on the nature of the computation that is happening in these regions itself. certain regions it might be possible to find lower dimensional representation, which is something we are assuming in our thing, right? We are assuming that we can break them down, the relevant information down to 20 latent dimensions. Now, whether the population activity itself is relevant or is it more dependent on single neuron activity is something that can definitely vary across different brain regions and might be the causative for the results that we see.
Okay, that's very interesting. So thanks again for the talk. Since it's 15, let's try to keep the time and moving to the next talk. Of course, if anyone has other questions, please feel free to write them in the chat and the panelists can take on them. And now our next speaker will be Ananya Passi from the Indian Institute of Science in Bangalore. And she will talk about the shape-sound associations, specifically, for example, why Kiki sounds spiky. I think it's a very interesting and curious topic. So Ananya, let's please talk about this.
Hello, am I audible?
Yes.
Okay. So, hello everyone. Today I'm here to talk about a very, very intriguing phenomena, that is the Kiki-Booba effect. Now, what is it? You see two shapes on the screen. I would ask you, associate one of them with the name Kiki and the other of them as Booba. So what would you do? Most often, people associate the spiky shape on the right as Kiki and the rounded shape on the left as Booba. But why is it so systematic? There are two competing hypotheses for it. According to the mouth shape hypothesis, when we say Kiki, our mouth forms an angular shape. And when we say Booba, our mouth forms a rounded shape. Thus, our brain associates angular shapes to the word Kiki and rounded shapes to the word Booba. The other hypothesis is the object sound hypothesis, which states that when angular objects fall, they produce a high-pitched sound. And when rounded objects fall, they produce a low-pitched sound. Thus, our brain associates spiky shapes to the high-pitched word Kiki and rounded shapes to the low-pitched word Booba. But how do we test these hypotheses? We could use reversed unpronounceable words to test this. If the mouth shape hypothesis holds, then both Kiki and Booba reversed words will not show the effect. However, if the object sound hypothesis holds, then they will show the effect, and most of the reversed Kiki words will be associated to spiky shapes, and the reversed Booba words will be associated to rounded shapes. So the first experiment involved the validation of the stimuli. Here an image was shown to the subjects and they were asked to associate one of the two names to it. These were the stimuli that we validated, and we saw that most of the spiky shapes were associated with Kiki-like words and the rounded shapes were associated with Booba-like words. And the results were consistent across subjects. The next experiment involved playing an audio recording and asking the participants to associate it to one of the two shapes. So these were the 6 different kinds of audio recordings that we showed. Takiti. Was this audible? Hello?
Yeah, we could hear it.
Okay. And so this was a pronounceable Kiki-like word, and this was its digitally reversed counterpart. Then we had environmental Kiki-like sounds. Then pronounceable Booba-like words. Lau. Their digitally reversed counterparts. Wal. And environmental Booba-like sounds. So, on playing these sounds, we saw that, of course, the Kiki-like and the Booba-like words showed the Kiki-Booba effect. However, the digitally reversed sounds also showed this effect, and so did the natural sounds. More interestingly, the responses to the pronounceable words and their digitally reversed counterparts was very, very correlated for both Booba-like words and Kiki-like words. Interestingly, there was a nice correlation seen between the frequency of a word and the Kiki response that it elicited. High-pitched words were more associated with spiky shapes, and low-pitched words were more associated with rounded shapes. This was the case for all pronounceable words, reversed words, and also for natural sounds. So, in conclusion, we can say that the Kiki-Booba effect is not limited to pronounceable words, and high-frequency sounds are associated with spiky shapes, and low-frequency sounds are associated with rounded shapes. Thank you.
Thanks, Rania. It was a very interesting talk. I've never heard before of this effect, so it sounds quite interesting, this coupling between what we see and what we hear and what we see. And if I can start with question, one question would be, do you think that the reason for this association is in some kind of neural representation? Is there a hypothesis behind it?
I'm not very sure about what part of the brain is associated with this, but there are people believe that the prefrontal cortex is associated with the cross-modal associations.
Okay, that's interesting. Do you think that are there any similar effects that has this kind of shape sound coupling?
Yes, a lot of what we speak has to do with the lip recording. McGurk effect is one of the examples. I could send you a link, and it's a very interesting optical illusion to see. So they're the same audio, but different lip recordings make us perceive the audio in different ways.
Wow. Oh, it sounds really interesting. Are any of the participants has any question, or the panelists have any curiosity on this talk? Let's see. So we have a question from Cleo Gergula. Very interesting, thank you. Do you hypothesize perhaps an evolutionary contribution resulting in this phenomenon of high-low frequency shape perception?
I think there is an evolutionary advantage of like cross-modal associations. For example, when you hear a sound, it is evolutionarily beneficial if you can have an idea of what shape produces that sound. So I think cross-modal associations will have a benefit, but high-pitch, low-pitch, I'm not sure.
Okay, do you think that it can be basically as the brain has been evolving, that associating something spiky that can be dangerous with a high frequency sound may have caused this association? We are just speculating here, of course.
I'm not sure about like danger, but I think it's useful in perceiving the identity of an object, right? I'm not sure of danger.
Yeah, and would it be also associated with the other senses? So here we are looking mainly at, we have a shape, is spiky, we see it, and we perceive the kiki sound. Then would you expect that there is also an association, for example, with the touch?
Oh, I'm not sure. Not sure. But I can tell you one thing. It is associated with other things. Like a similar experiment was done in which people were had to associate emotions with the shape, and they associated energy with the spiky shapes and like peace with the rounded shapes. So that's another thing. I'm not sure about touch. Maybe there is an association with like of pain with the spiky shapes and smoothness with the rounded shapes. I don't know.
No, that's interesting. And would you like to tell us if you have already plans on extending this study, or what would be your next step?
Okay, so we have done one more experiment as a control. I can show that to you. We wanted to know if the mean frequency was the only thing that affected the Kiki Booba-ness. So what we did was we created sinusoids of the same mean frequency as the words. Like booba, the word, I computed the sinusoid of that main frequency. And we asked people to associate it with the spiky shapes and the rounded shapes. And here also we saw that there was an association. So tones of the Kiki words had like were more associated with spiky shapes than the rounded shapes. And again, but there wasn't as much as the correlation as the pronounceable and reversed words like this. And it was a weaker correlation. So this is one thing that we did. Another thing that we want to do is we want to make a model, a sound shape predictive model that would be made on like trained on object images and the sounds they produce. And then we want to input object like Kiki Booba images into that object and see if they produce sounds that are similar to the words Kiki and the word Booba. This is one thing.
Yeah, that's very interesting. And if I can ask you a last question, and of course, if the participants or the panelists have other ones, we can also go with them. So you showed that in the bar plot that the actual words, Kiki and Booba, yes, basically they are very, very different. And then when instead you go to the tones, so the natural sounds that resemble them, they are still different, but they are kind of get closer. Do you think that instead of using real sounds, if you used just basically a sound based on a sinusoid of different frequencies, so basically a vibration, something like that, that it's not as complex as a real world sound, do you think that you would have something as the Booba Kiki difference, so such a striking difference?
We did this. So the tones, that the thing that you're speaking about, simple sinusoids, we just call them tones. So the tones did show a difference in the responses. Like the tones correspond, like high pitched tones were more associated with the spiky shapes, and low pitched tones were more associated with the rounded shapes. But the effect was not as pronounced as that of the complex words that we say, like Kiki and Booba.
Okay, then I guess that we rely on not just the main frequencies, but also on the harmonics that we produce with the words. Thanks again, Ananya. It was a very interesting talk.
Thank you.
And we can move on to our third talk from Lisa Knaus from the Institute of Science and Technology in Austria. And she will talk about the new conditional knockout mouse line to study microcephaly. Lisa?
Yes, I'm here. I'm just waiting.
Sorry, I'm not able to stop sharing my laptop.
I can do that for you, no worries.
Thank you.
Okay, so let me share my screen. Good. So we're shifting fields completely now. So I'm a PhD student at ISD Austria in the group of Gaia Novarina. And as already Matteo said, I'm working on a large neutral amino acid transporter and its role in neurodevelopment and in neurodevelopmental disorders such as ASD and microcephaly. So I'm working on a gene called SLC75 that encodes a large neutral amino acid transporter, and this is shown here in light blue. And this transporter forms a heterodimer with another protein, and this heterodimer is then referred to as LAT1. And LAT1 catalyzes a large neutral amino acid flux into different tissues, and among their substrates are also these essential branch chain amino acids. And it catalyzes this flux in a sodium and pH independent manner. So it's highly expressed in different tissues that have a high amino acid demand. Among them is, for example, the brain, which we are mainly interested in. So it's highly expressed in neuroprogenitor cells and in neurons. So why are we exactly interested in this gene? So a few years ago, we identified a subset of patients that harbor homozygous missense mutation in the SLC75 gene. All of these patients present with severe motor deficits. They are on the autism spectrum and they have severe microcephaly. So what we are now, in my PhD project, I'm mainly interested in the microcephalic phenotype of these patients. So we're trying to figure out which role SLC75 plays in neurodevelopment and how a loss of this transport is causing such a strong neurodevelopmental defect. But before I start, I just want to clarify some terms. So what is microcephaly? So in fact, the patient is diagnosed with microcephaly if the head circumference is 2 standard deviations smaller than average. And microcephaly can be divided into 2 subtypes. On one hand, you can find so-called primary microcephaly, meaning children are born with a smaller brain size, and this is usually due to defects during embryonic development, so defects in neuroprogenitor cells or in neuronal migration. However, there is also this so-called secondary microcephaly, meaning children are born with a normal brain size and start to develop microcephaly after birth due to mainly defects in processes involved in neuronal refinement, such as pruning or myelination. And of course, an increase in cell death of neurons or glia can, for example, contribute to this defect. So how are we studying microcephaly in the Novarino lab? I'm working with a conditional knockout mouse model of SLC75, where we're deleting the transporter in the developing cortex and hippocampus, more specifically in radial glia progenitor cells and their offspring, so excitatory cortical neurons and astrocytes, at a very early developmental time window, so at E11.5. What was quite curious to see was when, although we're deleting this transporter so early in development, these mice are actually born with a normal brain size. However, they start to develop postnatal microcephaly. So this you can nicely appreciate here in this plot, where you can see embryonic time points and early postnatal days after birth, and this is the brain weight, and you can start seeing that there is a difference in the brain weight starting to arise at around day 2 or day 3 after birth, which then gets really, really obvious at day 5 to day 6. However, after day 10, this difference stays constant, meaning what we're seeing and the effect that the loss of SLC75 has seems to be restricted to this very critical period after birth between P2 and P10. And this actually has been shown in neuron development to be a crucial time window where neuronal refinement is taking place. So the first thing that we of course did when we saw these changes in in brain size, we looked at general brain morphology. What you can see here is that these mice show a severe reduction in cortical thickness by around 40%. Looking at the cortical layering, you can see that especially the upper layers, so layer 2, 3 of the cortex, are significantly affected. So their thickness is severely reduced, whereas the lower layers here are affected, but is not as strong as in the upper layers, indicating that the layer 2, 3 pyramidal neurons seem to be more susceptible to the loss of SLC75. seeing such a severe phenotype, we of course, first of all, wanted to know what is happening to these neurons. So the first thing that I was checking is apoptosis. So what if these neurons just die? And indeed, this is what we're seeing. So I did immunoblots and immunostainings for cleaved caspase, which is like one of the main apoptotic markers. And this is a time course from P0, so birth, until adulthood. And what you can see is that we see a significant increase in apoptotic activity, again, during this very distinct time window where we're also seeing the microcephalic phenotype arising. What was quite curious to see is, so for people that are familiar with the literature on this postnatal period of neuronal refinement, this peak kind of is quite well known, especially regarding programmed cell death and neuronal refinement, because it has been shown in literature that in cortical development, so murine, but also in human cortical development, exactly during this time window where we are seeing the increase in apoptosis, so between P2 and P10, a wave of endogenous cell death is moving through the cortex, eliminating different cell populations due to, or to ensure a properly functioning network. So what has been shown by the marine lab is that actually between P2 and P5, you have first of all a wave of apoptosis hitting pyramidal excitatory neurons, and later between P5 and P10, exactly the window where we are also seeing the increased apoptosis, you see a wave of apoptosis hitting the inhibitory neurons, making sure that the cell numbers are adjusted again and that the ratios between the cell types are constant and a proper network is formed. So what has been shown, the factor which is modulating this cell death wave is actually neuronal activity. So neurons which are less active tend to undergo apoptosis, whereas neurons that are more active remain integrated in the system. So this was quite interesting for us to see, especially since we also see the wave of apoptosis in our mutant mice exactly during this time window, and we wondered if there might be a connection between a large neutral amino acid transporter and neuronal activity. And so we decided to do some electrophysiology. So this was done by a postdoc in our lab, Bernadette. She used current clamp recordings in layer 2-3 pyramidal neurons in acute slices exactly during this time window where we see the increased apoptosis. And indeed, what we find is that these pyramidal neurons in the mutant mice show a significant reduction in their firing rate. What was even more interesting to see was if we repeat these recordings at later time points, so during juvenilehood, this difference is again gone between mutant and wild type littermate control. So what this indicates is that we're really seeing a purely developmental phenotype here, and that it seems that SLC75 plays a really important role during this critical developmental time window of neuronal refinement. So another result that is nicely underlining this is shown here. So additionally, what I did is I looked at different other cell types which are present in the cortex, in this case, PV positive inhibitory neurons. What I have to mention here is that these PV positive inhibitory neurons are actually not affected by the loss of SLC75 due to the Cree line that we're using. However, what we're seeing is also that the inhibitory neurons are adjusting their numbers, so undergoing cell death, especially in the layers where we also see the majority of the neuron, the excitatory neurons lost. What this is showing us is that we're indeed exactly hitting this time window that was introduced by the marine lab, showing that the inhibitory neurons as a secondary effect are also starting to die to adjust their numbers accordingly. So due to time reasons, I have to sum it up already. So what I could show you today was that a deletion of SLC75 in excitatory forebrain neurons causes postnatal microcephaly. This is due to an increase in cell death during a very distinct developmental time window. What we also see is that during this distinct developmental time window, we're seeing a decrease in firing rate of corticopyramidal neurons, which might be even the cause why these neurons start to undergo apoptosis. So what remains now for me, the most crucial question I have to answer now, why do we see this reduction in firing rate? So what we're checking now is it due to the loss of the transporter itself, which might be modulating, for example, voltage-gated ion channels, or is it due to the loss of the substrates that the transporter is transporting? So yeah, with this, I would like to thank everyone that is involved in this project, and I'm happy to take some questions.
Thank you, Lisa. You made more than perfect timing, and it was a very interesting talk. So now it's question time, and I would like to start. So you showed that, can you share again your slides?
Yes, sorry, I'm just here.
No worries.
Okay.
You showed that slide where you had basically the histological section of the normal and the microcephalic brain. One next, yeah, exactly this one.
Mm-hmm.
So yeah, so the cortical changes that is evident, and you talked about it. What I wanted to ask you, what is known about instead what is happening at the subcortical level and in terms of the white matter connections?
So the thing is that we cannot exclude that something would also happen in subcortical regions. The thing is, the crystalline that we're using is just targeting the cortex, or the developing cortex and hippocampus. So this is something that we would have to check at one point. In parallel, we were breeding also with the nesting crystalline, which is deleting the transporter in the whole brain. However, this is something that we stopped at one point because the pups were not really very healthy, because we were just removing it from the whole brain. Regarding the corpus callosum, so the white matter tract, I was quantifying if there is a reduction in the corpus callosum thickness, for example. We didn't find any. However, I would, and this is on my list, also want to do some stainings or quantifications of MBP, so to check if there is a decrease in myelinations, just to be sure if this is also contributing to the phenotype, of course.
Yeah, sure. And also because in terms of volumes, of course, I imagine that this reduced volume is both if you look just at the cortical and the white matter, right? And also, I imagine that even in this case, that if I understood right, the transporter you are targeting is just for the cortical neurons, but you may have an effect also in white matter, given that it looks shrunk in the picture. Is that right?
Yeah. So this is something that we really have to check more. I mean, also considering that you lose a substantial amount of neurons which are projecting onto the different, so on the second hemispheres, it would be something that we would expect that you have also a reduction in the corpus callosum, for example.
Okay. So while I'm waiting for questions from the other participants or the other panelists, if they are curious about something, I was puzzled by the fact that basically the inactive neurons die while the active ones are still alive. Do you think that maybe it's a naive question, but is it something that you can exploit to react in some way to the pathology? There would be a way to make the inactive neurons more active over time in some critical period to make them survive?
It's a very, very, let's say, nice. So it's an idea that is really, really nice, especially in the part, like in the field where I'm working in. Of course, we're all trying to find some possible ways of treating these disorders. The problem with microcephaly is that you would have to treat already during, in most cases, you have to treat already during embryonic development, which is, of course, always a bit tricky, especially because these children are often diagnosed after they get born. But of course, it would be very, very a nice idea if this would be possible. However, with boosting activity in humans in the brain can be a bit tricky, because of course you can then induce some other effects that are going more into direction seizures, which you also don't want to boost too much. So I think this would be very, very challenging. But of course, if we find the modulator that is actually decreasing or modulating the activity, we can start to plan or think about to modulate this factor and not the activity itself. That would be the idea, of course.
Thank you. And to conclude, would you like to briefly tell us about how we could, how you could extend these results for therapeutic goals? How this could help to solve the issue?
So I didn't have time to show this, but we actually already found that, so SLC75 is also expressed at the blood-brain barrier. If you delete it there, then you will get mice that resemble more the phenotypes that I showed you before, like that are more like autistic-like phenotypes, social defects, and so on. So we were able to kind of treat these mice by injecting branched-chain amino acids, so the substrate that SLC75 is transporting into the brain. So this worked very, very nicely and was very, very convincing. However, if we're now seeing that the phenotype here is coming from the neurons, this will be very tricky because injecting the substrate into a cell type itself will be quite challenging. So this is maybe a bit tricky, but there are possibilities how you can approach this.
Okay, thank you very much for the answers. And since it's 45, we made perfect timing, and it was a very interesting session. Thank you to all the speakers, and for all the participants, take a 15 minutes break, and I'll see you in the next sessions. Thanks again, everyone. Aha. Okay, and I guess we are live, and we managed just in time to get all our speakers. Good morning for the early risers on the east coast of the US, and almost good afternoon for people from Europe. We have a very interesting session coming on. It's mostly on electrophysiology on different levels. We will go first through EEG, MEG stuff, and then we will go deep in the actual invasive recordings in macaques. And I guess that I talked about nonsense for a bit, so I can move on. Of course, feel free to ask questions both here and on YouTube, and we will answer those. If you are surprised by my face, it's just the Halloween competition. So our first speaker is Ryan Timms from the University of Oxford. And we'll talk about TASER. We have another one of these nice acronyms today. And in this case, TASER is an unsupervised algorithm for EEG, MEG source reconstruction. Very curious about it. Tim, Ryan?
Thank you very much. Can you see my screen? Can you see the screen?
Yes, sorry, I was muted. Yeah, we can see it.
Okay, cool. Thank you for the introduction. It's really nice to be here, and I hope everyone's had a good conference so far. So yeah, my name's Ryan. I've been working with Mark Woolrich for a couple of years now on a new approach to MEG and EEG source reconstruction, and that's what I'll be talking about today, and it's called TASER. So source reconstruction is a really key step in MEG pre-processing and can improve the utility of the data that we measure. But to actually succeed in carrying out source reconstruction, we must first solve something called the ill-posed inverse problem. And this basically arises from the fact that typically we reconstruct thousands of sources from hundreds of sensors. And to do this, we model the source space currents as a linear sum of the sensor level recordings. But to actually derive these reconstruction weights, we have to make some a priori assumptions about the data covariance. More elaborate source reconstruction techniques take a data-driven approach to this. But for the most part, all of these approaches introduce the sometimes oversimplifying assumption that the covariance itself is actually static in time. But our intuition tells us this can't be the case. We know that the brain is highly dynamic and exhibits changes at the millisecond timescale. So to pivot another way, this is like us watching a movie which is tens or hundreds of minutes long, and then averaging each of the individual frames of the data together to get to representation of that movie. And this isn't wrong per se. This is a valid representation, but it's not the best representation. As you can see here, the average of all of the frames doesn't look anything like any of the individual frames at individual timescales. And this is typically what's happening with MEG source reconstruction, where we collapse in time. So you might now ask, well, if I've got two experimental conditions A and B, can't I just manually segregate these conditions out and construct covariance windows from these different trials? And indeed, you can do this in more simplistic experimental setups, and it's a valid way to proceed. However, this doesn't take into account any neuronal intertrial variability, and it typically makes the assumption that trials are temporally aligned in time in terms of neuronal response. But also it gets problematic when you have complicated experimental setups and you might have more than two conditions, and being able to say specifically when a trial or when a neuronal response takes place becomes hard. And finally, we're still collapsing several hundred milliseconds of data here into one time point and using one or two reconstruction weights. And this problem gets worse when we move to the resting state, where there is no trial structure at all, and it becomes hard to say when an event takes place. But also, if we've got a complicated paradigm, like someone's watching a movie, drawing the lines then as to when condition A and when condition B is really hard. And sometimes we do have some underlying temporal structure, but it can be hard to manually pluck out when this is, for example, with epilepsy data. And so this is why we make the argument that we need an unsupervised, temporally adaptive approach to MEG and EEG source reconstruction, where an unsupervised algorithm can pluck out interesting segments of time in the data and construct covariance windows from these time points. And this is what TASER attempts to do. And in the TASER algorithm, what we do is we model the time-varying covariance as a weighted linear sum of basis functions. These dynamics, the temporal element of this, are taken into account by these alpha coefficients. And these coefficients are parameterized by a recurrent neural network, which basically learns a mapping from the raw sensor level MEG and EEG recordings to the alpha dynamics. And then the spatial covariance is encoded with these beta basis functions, as shown here by the colored blobs on the brain. And once we've got this estimate for the time-varying covariance, we can then have time-varying reconstruction weights for every time point in our required data. And this is all a very nice idea, but to actually show that it's worthwhile doing, we applied TASER to a pre-existing data set where subjects were sat in an MEG scanner and were asked to tap their index finger and pinky finger sequentially. And if you don't model the data at all, or you assume that the data is static and you reconstruct this activity, we are not able to distinguish between index finger and pinky finger activations with MEG data. However, if we apply TASER to the algorithm and separate the trials in an unsupervised way, I should stress, between index and pinky finger activation, we are able to distinguish spatially the difference between between index and pinky finger motion in MEG. And so hopefully with that, I've made a somewhat convincing argument that TASER is a tool which we need to employ to really get the best out of MEG and EEG data in an unsupervised fashion in a range of different tasks. And with that, I'd like to say thank you to my friends and collaborators who've been really helpful so far with all of this work, and for you for listening.
Thank you, Ryan. It sounds very interesting. I worked with the source reconstruction in the past, and I remember that the problem is that basically you cannot sometimes trust your results, or you will have these problems where if you want to identify find differences, it will be kind of hard. So is there any drawback in the method you propose? Of course, this was very brief, but it seems too good to be all true. Not that it's false, but is there something that it's challenging for Taser?
Yeah, so it's a very good question. I should say that the usual rules apply in terms of covariance, estimation, and accuracy when you're trying to be dynamic. So if you don't have enough data points for an event, or if your signal to noise ratio is too low, you will still suffer from the same kind of drawbacks which a conventional beamformer or multiple sparse priors algorithm will suffer from, because you need that data to be there. So that's one thing. And then I guess, as with all source reconstruction algorithms, when you move to real data, there is no ground truth. And so your concern, I guess, will always be there. Yeah.
Thanks. And we have a question from the audience. So it's from Jules Brochard. If you alter the timing in your data, would you risk to confound the cause from a consequence? For instance, the sensory motor perception of your finger.
Sorry, could you repeat the question one more time, please?
Yeah, sure. So if you alter the timing in your data, would you risk to confound a cause from a consequence? For example, the sensory motor perception of your finger rather than the actual finger movement?
So just to clarify, we're not actually altering the timing of our data here when we run Taser. So we don't manipulate the order of events or anything at all. We get a reconstruction weight for every single time point of the data set which we feed in. But to have a stab at a question, which I think is a more general one, yes, if you're not careful, your conception, if you conceive that you're about to tap your finger, that can sometimes confound your results. So you have to be careful with your experimental design and your baseline period. I don't know if that answers the question or not.
We'll see if, Jules, if you want to come on stage or if you want to ask a clarification, feel free to do that. And in the meantime, I wanted to ask you, for applications of Taser to EEG data, would it suffer from the same issue of normal source reconstruction algorithms, where you need to have a decent amount of electrodes, or can you in some way, do you have some advantage over that, and can you afford less electrodes than in the case of classical reconstruction algorithms?
It's a good question. The short answer is I don't know, because I haven't done that investigation yet. But yes, I would suspect that you will suffer from the same kind of classical drawbacks. And to keep, say we had a set number of electrodes, you won't get better data quality. Your signal to noise ratio in your raw recordings won't change. However, the confounds which are introduced by assuming that the covariance is static, you would see an improvement there. So the raw data won't get any better, if that makes sense. But you might glean some extra information by being intelligent with the dynamics of the covariance. But I would have to set up the simulations to give you a concrete answer.
Okay, thanks. And Richard, Etienne, any last question?
Yeah, following up on that last point, it seems like having a method for estimating changes in covariance over time is just a very general and powerful method. And I'm curious if you thought about applying that part the algorithm to other data. For instance, I mean, I'll be talking later a little bit about whitening voltage data from electrophysiological recordings and how that kind of helped us. But that, a core problem of that is getting a good estimate of a covariance matrix, which I usually assume is static, which I know is not a good assumption. So have you thought about applying that part of the toolbox to other data?
To be completely honest, it's kind of happened the other way around. So my supervisor, Mark Ulrich, and his students in the past have really kind of cut their teeth in modeling covariance and time-varying covariance with a thing called a hidden Markov model. And I would agree with you that it's a kind of more general problem to solve is modeling this covariance. It just so happens that source reconstruction, when you take a data-driven approach, is all about the data covariance. So it's a really nice natural application of these modelings of covariance. Very good question.
Okay, and if there are no further questions from the panelists or the participants, we can move to our next speakers. Thanks again, Ryan. And the next speaker is Etienne Cobrisson from the Institut de Neurosciences de la Timon. And I said before that today it's all about acronyms, and he will present FRIT. I hope that my French is good. A hierarchical functional connectivity approach.
Yeah, it's good.
Etienne?
Can you hear me?
Yeah.
Okay, and do you see the slides?
Yes.
Okay. So thank you, Matteo, for the introduction. So indeed, FRIT, which means Framework for Information Theoretical Analysis of Electrophysiological Data and Statistics, is a Python software I started to develop last year and is hosted on GitHub if you want to take a look at it. So if I have to summarize what is FRIT for, I would say that first it's really made for electrophysiological data, so EEG, MEG, and intracranial data. And the main goal of FRIT is to try to extract connective networks that are modulated according to the task. And this extraction, we try to do it within the information theoretical framework. And we also have in mind the possibility to draw statistical inferences either at the subject level or at the group level. So an illustrative example, so let's say you have 3 brain areas. The first level of analysis that you can perform within FRIT is ask the question whether there is some brain areas that shows some neural activity that is modulated according to the task. So it can be, for example, condition A versus condition B, or a regression against a model or whatever. The next question you can ask is whether the connectivity between the 2 nodes is also modulated by the task. And the ultimate goal we try to achieve within FRIT is asking the question if the flow of information starting from one brain region to another is also modulated by the task. So if you summarize a little bit the method that we usually used in neuroscience, it's mainly correlation, t-test, decoding, partial correlations. And there is this really nice paper of Robin Ince, which show that most of them have an equivalent in information theory, especially using a versatile tool of the mutual information, which measures the statistical dependency between 2 variables. So this is a really nice framework because you can really work inside this framework and do all of your analysis using almost the same tool. So within FRIT, what you can do is multi-level analysis, statistical inferences. So for example, you can perform statistic at the subject level and know how many subjects have the effect. You can perform fixed effect analysis where you try to extract some typical characteristic of your population, and you can perform random effect analysis where this time you try to build a model of how your mutual information is distributed across the subject. And this can be applied either at the node level, so really on the brain areas, or on the connectivity between brain areas. So a small example, now we are trying to validate all of the method and the statistics. So it's a visuomotor task, and there is the paper of Andrea Brovelli. So in this task, the subject on the screen, there is a color that is displayed, and the subject, they are learning finger association with the color. So at the beginning, they are really exploring which finger correspond to which color, and at the end, they know the association, and they really exploit this association to get the reward. So the first question you can ask is whether there is some brain areas that shows differences between the exploration and exploitation phase, and we retrieve results from the paper where there is big differences in occipital lobes. So I forgot to say, but this is computed on high gamma activity estimated on the MEC data. And another small example, but the connectivity, especially inside the occipital lobe, is also really different from the exploration versus the exploitation phase. So I guess it's almost 5 minutes. I don't really have the timer right now, but here is the link to the software if you want to give a shot, and here is the people I've been working with to develop it. So thank all of you for your attention.
Thank you, Etienne. You made perfect time by yourself. I didn't show you the cards because I didn't have the chance to show in them before, so I didn't want to confuse you, but you were in perfect time. And
okay, so perfect.
We have, I guess it's a small question in the chat asking which paper made the translation from statistics to mutual information. Do you have any idea of which one would be that?
Which paper make the translation statistic to mutual information?
Yes.
I don't really understand what you mean, Jules, by the translation statistic to mutual information. Because maybe at one point I say that your mutual information measures the ones. Okay. It's the paper from Robinins 2017. And let me know, it's this one, a statistical framework for neuroimaging data. It's a really, really nice paper where he shows the equivalence between some tools and some with the mutual information. And overall, it's a really nice introduction to information theory, and yeah, really, really cool paper.
I have a question at the end about mutual information. So when you work with the image alignment, that it's kind of different with working with the electrophysiological recordings. Mutual information is both a powerful tool, but also a damnation because it makes you align images that are very different, but then you may be unlucky and the results would be a mess if the relationship is nonlinear. And since here you are basically matching group level and single subject level recordings with the mutual information, did you have any issue in which you could have some problem in matching the two? Did you have to overcome any challenge using mutual information?
Yes, so one big challenge with the mutual information, and this is a really good question, is usually it's performed with a binning. So you have to bin your data, and usually it requires a lot of data to estimate a true covariance matrix. So in the paper of Robinins, he introduced a version of the mutual information, and I think it's originate from economics, in fact, where it's using what is called a Gaussian copula mutual information, in which you don't need binning. It's a binning-free method. However, it also means that you should have a roughly monotonic relation between your two variables. So if it's roughly increasing or roughly decreasing, it's okay. But if you have, like, for example, a curve, x square relation between your two variables, in that case, it's probably not going to work. So this is one limitation, but yeah, overall it works quite well, and I think it's a relatively okay limitation, I would say. It's a good compromise, I would say.
Okay. And to follow up, we have another question from Jules Brochard, that is, can you elaborate on the connectivity criteria Frit offers? How do you infer a complete graph?
So inside Frit, we have several measures of connectivity. We have non-directed connectivity, but all of them are single trial. So it's basically computing the mutual information between two time window and between two areas. And we also have covariance-based Granger causality. So in that case, it's directed connectivity. And we have a univariate version, which it's a bivariate version, sorry. So you really look at pairwise connectivity. And there is also another package which is written xFrit, but pronounced crossFrit. And in that case, we are testing a multivariate covariance-based Granger causality. So yeah, currently it's in testing, but I think the results are quite promising. So Jules, if you want to discuss, we can talk about that if you want.
So to follow up the question, I don't know if Ryan first want to ask a question just because he unmuted.
Yeah, I was just going to ask, is there any particular reason why you presented the gamma activity results here? Was that of particular interest to you, or did you look at other bands as well?
No, okay, so in the example I show you, so it was this one. It's really high gamma activity because the paper that we are trying to reproduce and validate the method is using high gamma activity. However, Frit is also compatible with frequency temporal investigations. So you can pass full time frequency map and see if there is time frequency clusters that are different between conditions, for example.
Do you think you're potentially going after quite a hard thing to show here with the gamma band stuff?
Sorry?
I know that some researchers struggle to get reliable gamma activity results.
Okay, you mean gamma estimation with make data?
Yeah.
Okay, so for that, I don't know because I didn't make the gamma estimation. It was really the work of Andrea Brovelli and his paper. Okay. And now me, I'm more using intracranial data. I'm way more familiar with that.
Oh, right. Okay.
But I think if you really want to dig into it, there is a repo inside our project that is called, how is it called? BV2MNE. And in this one, there is a gamma estimation, but I don't know so much the details, so I don't want to say something wrong to you.
Okay, cool. Thank you.
Yeah.
And if I can follow up on what you were saying on Frit and cross-Frit. So I imagine that then with the Frit, you wouldn't be able to discriminate between indirect and direct functional connectivity, in the sense that if you have three areas basically, and two are connected and the third one is connected to the other two, so there is a case that you will have a functional connectivity because of an indirect connection. With cross-Frit, you should be able to identify that difference. Should you?
Yes. Yeah, so exactly. So the problem with the Granger causality is that, for example, if you have a 3 nodes and you have node 1 that is sending to node 2 and node 3, you can have a spurious connectivity from node 2 to 3.
Yeah.
So in that case, this is one limitation of the bivariate case. And in that case, you need a multivariate Granger causality. So this is what we are currently exploring inside cross-Frit. And so, okay, cross-Frit is in fact experimental features of Frit. that are not included for the moment inside FRIT, but it's still open, so you can take a look at it. And in that case, this version, multivariate version, for it. And I also have to say that the covariance-based measures not only gives you if one brain area is sending information to another, but it's also giving you the information if two brain areas have an instantaneous connectivity together that cannot be explained by the past. So it's a complex measure, but it's really interesting, I would say.
And any final comment about the pitfalls of Granger causality in the fact that some people will argue that it's not causality per se?
Well, it's true. I mean, I think that's why usually the people say that it's causality in the sense of Granger. So I think it's a good precaution. I don't know if it's causality or if it's just delayed activity. It's hard to say, but at least it also means that you can find and estimate the delays between your brain areas and see if they are instantaneously activated or if there is a delay between them, and how is this delay propagated across your brain areas. I think it's a safer assumption to say that.
Yeah, it's a pity that we don't have Conrad Cording in this call, because we would have a fun time then. But no, thanks for the answers, and thanks for the talk. It was very interesting. And now we will move to our last speaker, Richard Lange from University of Rochester, that will have a surprise Halloween costume in a bit. And he will talk about a study on the macaque visual cortex where they use both electrophysiology and histology. I really look forward to that. Richard?
Yes. Hello, everyone. I wanted to add an acronym so I could join in with the rest of the session, but moving on. So just to start, I want to acknowledge collaborators and funding upfront, because this was very much a group effort. So there's everyone. So our goal with this project is to further understand the relationship between a neural population sensitivity to a stimulus, neurons' choice probability, and correlated variability between neurons, as well as how the latter two depend on feedback to sensory cortex. Linear decoding models predict that choice probability depends both on neuron sensitivity to the stimulus as well as on the correlated variability. And indeed, numerous previous empirical studies have reported an association between choice probability and sensitivity. Other studies have found task-dependent structure in noise correlations, suggesting that feedback must be involved. And these task-dependent changes in noise correlations are not easily explained by any feed-forward decoding framework. In separate theoretical work, we were able to derive the optimal form of feedback to sensory areas by assuming they are involved in the hierarchical inference process, where feedback communicates prior expectations. One goal for this theory was to give a unifying account of why F-prime structure appears in both choice probabilities and in noise correlations. So to briefly summarize the entire work, we set out to test this theory and to extend previous experimental results on feedback-induced F-prime structure in choice probability and noise correlations, but we ended up failing to replicate the main effects, which raises questions about methodological differences as well as idiosyncrasies between brain areas, neuron types, or even monkeys. So over the next few minutes, I'll tell you about the experiment design and analysis methods we used to arrive at this failed replication. So we recorded from macaque V1 Utah arrays while monkeys learned two coarse orientation discrimination tasks. The first is a cardinal task in which they discriminated from horizontal from vertical patterns. And the second was an oblique task in which they discriminated diagonal patterns. An important feature of the stimulus is that it's identical at zero signal for both tasks, allowing us to isolate the effect of changing the task cue while holding the stimulus constant. We repeated the same training protocol for two monkeys, monkey U and monkey A. Both monkeys began learning the cardinal task and switched to learning the oblique task. They then learned to randomly switch between the two tasks based on a cue at the start of each trial. Monkey A underwent the entire protocol twice, once in 2017 and once in 2019. We analyzed data from between 5 and 20 sessions at the end of each experimental epoch, once the monkey's behavior had stabilized. Both monkeys did learn to perform the discrimination tasks well. Here I'm showing psychometric curves for monkey U during interleaf sessions. Monkey A similarly learned the task, but with less overall sensitivity and more biases, likely due to the more peripheral stimulus. In separate behavioral analyses, we found that monkey A did not significantly switch his strategy based on the cue, but only in 2017. So in the interest of time, I will move on to neural data. Neural data were collected by 96-channel Utah arrays implanted in V1. And unlike previous studies, we decorrelated voltages across the different channels by ZCA whitening and found that this dramatically reduced artifacts and correlation estimates. For further analysis, we counted all spikes per channel in a window after stimulus onset. We did not spike sort because all previous studies on this topic have reported no difference between single and multi-units. We then fit a non-parametric statistical model to spike counts across trials, which we use both to subtract off slow background fluctuations and for automatic outlier removal. In the end, this only removed a very small fraction of the data. In the end, our neural data appeared pretty much as expected in terms of orientation tuning, PSTHs, tuning to the signal in the task. Here I'm just showing a few example neurons. And noise correlations were likewise consistent with previous work, both in terms of average and in terms of distribution. So turning to the first main analysis, we regressed choice probability against normalized neural sensitivity. While standard regression methods typically assume all data points are independent, this is not the case for our data because we used chronically implanted electrodes. So we used hierarchical Bayesian regression to correct for the correlated measurements. Essentially, this performs separate regressions per session and per electrode and stitches the data together. In the example shown here, we see a weak but significant correlation. But when we look at the rest of the data set across both monkeys and both task types, we see no significant relationship between choice probability and sensitivity consistently. So to summarize a few key points, for chronic array data, it's really important to account for the fact that measurements may be duplicated when stitching together data across days. We addressed this using hierarchical Bayesian or mixed effects regression, which is really important to avoid inflating significance. And second, we do see some interesting structure in here, but I've just been told my time is up, so I'm going to skip that part. We had a second main experimental test about whether or not we see F prime structure in the covariance matrices based on projecting the covariance in each task onto the two axes. Long story short, we also found no results there, but in the interest of time, I won't describe these plots. So to summarize, hierarchical inference predicts task-dependent F prime, so sensitivity structure in both choice probability and noise correlations, which is supported by evidence from previous experimental work. But we failed to replicate these previous findings, which may or may not be a null result for the theory. More generally, this failed replication calls into focus differences with those previous studies in terms of differences in analysis methods, experiment design, idiosyncrasies of the monkeys, or even some unlucky sampling of the neurons. So thanks everyone for listening, and I'll be happy to take some questions. I'm going to leave up some FAQ and discussion points here. And if you give me one moment, I will get my entry for the costume contest, which is something that is both frightening and topical. So here you go.
What does it say? Okay, I'll just stop for a second your screen sharing just to take a screenshot because it's too fun. So Richard, thank you for the talk. Let's start with the questions. I have a question. So since one thing that I think you mentioned that it was different from previous studies is that you decided to use the whitening. Yeah. And it was effective in reducing spurious, in reducing artifacts.
Yeah.
Have you tried to use something different and something less effective to see if actual previous results may actually be based on including also artifacts in the analysis?
Yeah, I mean that's where we are on this. Is we tried both. We tried not whitening and we tried whitening, and I can tell you on our data, the main difference is sorry, let me find this slide. So yeah, what we find is that this reduces the number of like millisecond level coincidence spikes that we see across channels, which would be totally unexpected given the array spacing. So there's some additional like overall fluctuation that's shared between them. And I can point to this paper by Schultz et al. In 2015, which found that the single biggest contributor to correlations in this type of array was shared local field potential fluctuations. But in that paper, they did not try any methods to reduce those correlations. And what we've done is whitening, which we find empirically gets rid of those coincidence spikes across channels.
Okay.
We haven't done something more systematic than that.
Yeah, no, it's in any case, it's interesting that here, since your results look quite clean, it's interesting that then you get something very different from the literature. And of course, if the participants or the panelists want to jump on, please do. In the meantime, you mentioned in the abstract also histology, am I right? Did you skip it for?
Yeah, that was for time.
Okay. No, just because I'm curious on how you used it.
Yeah, so this is a brand new and unpublished experimental method that Rick Bourne is working on. Basically, they were able to cool down the cortex to the point that they could extract the probe and get an estimate through slicing of where the tip of the probe was. And what we found was that in monkey U, the majority of the neurons were in layer 4, where like the theoretical predictions, and I think what most people have in mind when they look at feedback in noise correlations and things, it's layer 2, 3. We don't yet have the histology data for the second monkey though, so we can't say too much more about that. Oh, and the other thing I can say is, even though the predictions are more likely to hold in layer 2, 3, if we just rerun everything on the layer 2, 3 neurons in monkey U, we still see null results.
That's quite puzzling. How did you, so I imagine that as you got these results at first, you would have thought I probably have done something wrong, right? Did you do any troubleshooting to see, to actually be convinced that there may be something bigger than just small issues?
Yeah, this has been a long process of convincing ourselves that there's nothing here, and part of the problem is this one data point. We have essentially a very small fraction of the data that looks exactly as expected, but everything else is just kind of not there, despite no other apparent differences between those sessions. And so we've applied lots of different analysis methods. We've tried direct replications of previous studies that I don't really have time to go into now. But you're absolutely right that it did take us a long time to realize that this ultimately is a null result.
So did the previous studies looked at a similar amount of monkeys and a similar amount of channels per monkeys, or?
Yeah, so on this discussion point, so should we even expect to see choice signals in V1? There are basically 2 previous studies, each with 2 monkeys, because that's the standard. So 2 studies that say yes, there are choice signals for this type of stimulus in V1. Hendrika Nienborg has a separate paper where she uses a different type of stimulus in V1 and finds no choice signals. It might have to do with the stimulus, but we use the same stimulus as these 2 papers. But there was another recent paper by Jasper et al. In 2019, which also, I think, used a similar stimulus, don't quote me on that, but did not find choice signals in V1. So currently, if you account for our study, that seems to be a tie for 2 yeses and 2 nos on whether or not these should be there.
Okay, no, that's interesting to hear. And actually, Etienne, if you want, of course, you can just ask Richard.
Yeah, I have one small question for you. What do you use to build your hierarchical Bayesian model? You're using R to make it?
Yeah, well, we use Stan, which
Okay.
yeah, which everyone who uses R uses Stan, but we put Stan inside of a MATLAB pipeline. So ultimately, it was a Stan model for the hierarchical regression.
And do you think a Bayesian model they can easily be used with time series data? I'm really not so familiar. I'm reading about it because I think I would like to switch in the future, but for the moment, it's not really clear to me how to use it. For example, with the time series data, say for example that I want to find if there is an effect somewhere. Is it possible to use this kind of model?
What kind of time series data?
For example, electrophysiological data.
Yeah.
For example, because for the moment, what I can have with my p-value is I can say if it's significant, but I cannot say if there is an effect or if there is no effect like you will have with the Bayesian factor. So that's why I'm more and more interested into this and to draw more reliable statistical conclusions. That say, I'm not sure how it works with time series data.
Yeah, I mean, okay, let me say one more thing about this. This is kind of a non-answer, but maybe it will help a little. So there's a lot of data processing that goes from the raw data to the points that we end up regressing, right? So we had, so we have this regression here, but each one of these data points reflects a large number of trials for a single neuron, which involves slicing out the right trials based on signal strength and all this stuff. In principle, it's possible to build one big statistical model that goes all the way down sort of to the raw data. Instead, what we do is we pre-process the raw data to get these XY coordinates in this space, and then we apply the statistical model on top of that.
So it means that you might are losing some information at the single trial level to extract some statistic about your data and then build your model on top of it.
Yeah, so the way we address that is, I mean, we have an estimate of the individual uncertainty in the X and Y coordinates of each of these individual points, which we get just by bootstrapping the data. So by resampling the trials, we can estimate our uncertainty in each measurement, but in the end, we're applying the statistical model just to the two-dimensional regression instead of the high-dimensional like time series data. Because my experience is that Stan is great for this kind of thing, but it requires much more hand-holding, if you will, to apply it to a larger, richer, time series data.
Okay. Thank you.
Okay, so it's 46, so we made good timing. I think it has been a very interesting session. We had 3 wonderful talks, and thank you for all the speakers. Thank you to the participants, to my back host, and enjoy the 15-minute break and enjoy the next sessions. Bye, and good afternoon.
Ciao.
Thank
Ciao,
you.
everyone.
Bye.
Welcome, everyone. Today we're going to have 3 very great talks, all focused on detectors and Hebbian learning and also synchronization models. The first talk is going to be by Abul Fazl Zia Emir. He's going to talk about the effect of anti-Hebbian learning on the synchronization and structure of directed networks with pure and hybrid inhibitory and excitatory couplings. He is a researcher in Dr. Mina Zaray's lab at the Institute for Advanced Studies in Basic Science, all the way from Iran. Take it over, Abul Fazl.
Yeah. Hello, everybody, and thank you for introduction. I'll go a very brief introduction about synchronization, and then the main question is what's the optimal structure for synchronization of a network at the presence of noise and heterogeneity. We introduce DAG network, directed acyclic graph, use Chromato model, and anti-Hebbian learning. or non-Hebbian. Synchronization is very common phenomenon in nature, industry, everywhere. People are studying synchronization for many decades. A very common usage in neuroscience is, for example, people use tractography to extract the graph structure of the brain, and then use some computational model to reproduce the oscillation in brain. For example, here we have some oscillation in EEG recording from human brain, which is it has a prominent algorithm, and use this oscillation and calculate the functional network compared with empirical data and something like that. Okay, the main question here, we are looking for the optimum structure for synchronization of a network at the presence of noise and heterogeneity. By heterogeneity, I mean, for example, oscillator have different frequencies. Previously, we know that the optimal structure for synchronization is directed acyclic graph, and it can also be proved mathematically that directed acyclic graphs, something like this, is the optimal structure for synchronization. This kind of network has a leader or hub, and it's connected to other nodes by feed-forward loops. On the other side, we have a balanced directed graph, it has many feedback loops. For example, here, if you see my pointer, we have some cycle or feedback loops here, and we have feed-forward loops. But directed acyclic graph, or in short DAG, are not the optimal structure, and even they are the worst choice at the presence of heterogeneity. Here, for example, if we have noise, which order parameters drop very fast, and also if we change the frequency of hub, the order parameter of the system drop very rapidly, and so it's not an optimal choice. We are going to solve this problem in this kind of network. We use chromato model. Theta is a state of each node at each time step. Omega is frequency. K is coupling. N is the number of node AIG elements of adjacency matrix. And this is the coupling term of a system. If I'm going to explain here the non-Hebbian rewiring. For example, we have some node here. In each rewiring step, we calculate this function, which calculates the phase difference between each pair of nodes and remove the connection between two nodes that have the minimum value of, for example, Fij, and randomly connected to another node. If we just apply this simple rule to a random network and with, for example, a sparse network and random, and start the rewiring here, it rapidly increase the synchronization of the network. By synchronization means, I mean that phase of all nodes are the same or almost the same. And at the end of the rewiring, we have a network with higher value of the order parameter or synchronization. If we just look at the distribution of in degree and out degree of the nodes, before and after rewiring, the out degree doesn't change, but in degree has a more uniform distribution. If we plot the distribution of degree versus the frequency, we have some shape like that for in degree of the network. And Another panel showed the distribution of average frequency of the neighbors versus the frequency of each node. These two panels tells us that each node with higher frequency tends to connect to the nodes with lower frequency. Okay. If we just look at the adjacency matrix of the network before and after rewiring, and also the correlation matrix before and after the rewiring, it also confirmed the previous result at the previous slide. And these nodes are arranged from a low to high frequency. For example, node with index 1 has the lowest frequency, and 500 has the highest frequency of the network. And correlation also somehow follow the structure pattern somehow. We can count the number of motif in network. For example, we can count the number of cycle or feedback loops in a network before and after rewiring. We see that the number of feedback loops drop rapidly for our hub. For example, here is our hub with frequency 0. Average frequency of the network has the lowest number of feedback loops. And if we look at the average shortest path of the network, we see some distinct pattern. If we are going to go backward to the hub, it has the most distance of the network. For example, if you are going to reach to the hub in a backward. Here I have counted the number of feed-forward loops. The number of feed-forward loops for the hub slightly increase, and this is what we conclude that from two previous slides, that the optimal structure for a network after non-Hebbian rewiring is the network that have average frequency for the hub and lower frequency and higher frequency nodes try to connect to the hub with feed-forward loops and connect to each other with feedback loops, as we saw before in, for example, in adjacency matrix. If we apply the same rule in a network with inhibitory and excitatory nodes with a fraction of 20 to 80, the value of synchronization doesn't increase as before, but we have a balance in degree between excitatory and inhibitory nodes, and also we don't see the distinct pattern as before for the just excitatory nodes. That's the difference between the two structures. Okay, the conclusion is that directed acyclic graphs are not optimal structure at the presence of noise and heterogeneity. To solve this problem, we used anti-Hebbian learning, anti-Hebbian rewiring, and find a structure that have the optimal structure. We see that nodes with frequency near the average try to connect to the hub with feed-forward loops, and the node which has higher frequency and lower frequency than the hub try to connect with feedback loops. I thank to the other collaborator, Minazarei. Thank you very much.
Thank you, Abul Fazal. That was actually very clear. We have one question in the chat. Jules Brochard says, naive question here, is there a physiological relevance, or sorry, physiological evidence for anti-Hebbian learning? What would be their purpose?
Yeah, there are many empirical evidence that showed in brain we have both Hebbian and non-Hebbian rewiring for learning, for example, in the brain. And I didn't bring here any reference. Yeah, I should bring some reference for empirical data, but we have some non-Hebbian learning at the occipital, for example, part of the brain that have been proved in experimental, for example. But there are many good review for yeah, finding both type of Hebbian and non-Hebbian learning the brain.
Thank you. And also, I was wondering if you take into account any reciprocal connections in this model. Are there, you know, like the hub, your hub network connected to and from the other, either the high frequency or the low connection probability network?
I don't know. I think it's I don't know. I get the question clearly, but can you repeat? Sorry.
If there is a reciprocal connection, so if
Yeah. Yes, because we are just rewiring in randomly, we are counting the number of, for example, feedback loop and feed-forward loops. It can happen many reciprocal connection, but on average, the number of feedback loops, for example, increase for high frequency and feed-forward loops, for example, in average frequency. Yeah.
All right. Well, I think that's it for the questions. Let's thank Abul Fazl for that talk. It was really insightful. All right, next we have TB Tesilanu. He is an associate research scientist in the lab of Dmitry Chilkovsky at the Flatiron Institute in New York. And he's going to be talking about biological learning of local motion detectors. TB, the stage is yours.
Okay, hi everyone. Yeah, so this is work done with Alex Genkin from NYU and Dmitry Chilkovsky at the Flatiron. So I'm going to talk about how to build neural circuits that learn local motion detectors. And in animals from fly to mouse, we have these motion sensitive cells like these retinal ganglion cells that respond to objects moving in some directions but not others. These are spike trains, and we see this moving down exhibits no response, moving up exhibits a strong response. And it also only works in a localized area on the retina. And for the fly, we have similar cells that respond to some direction of motion, but not other. And in both of these cases, the mechanism does something like correlate the signals from two nearby points on the retina with some different temporal dynamics. So if we think of this baby Yoda dropping an apple, and we have two receptors at 2 points on the apple trajectory, we have two cells, one with a slow response and one with a fast response. And by correlating the slow response from the upper cell with a fast response from the lower cell, we can detect motion that goes downwards. So this sort of mechanism is localized, I guess, in two senses. One is that it detects motion only in this area of the visual field, and another is that the connectivity is local. So this cell doesn't try to sample positions from very far away. So we can actually try to wonder why the detectors are localized. So we might imagine that if motion was global, it might actually be useful to try to pull together information from distant locations on the across the visual field. So a possible answer is that it's an adaptation to the fact that natural motion is not global. Motion is usually different in different places across the visual field because there are objects that are moving and objects are of different sizes. So the thing that we're wondering is whether we can learn motion detectors that are localized, like the ones we see in biology, from an optimization problem that uses moving images that are natural or naturalistic in some sense. And the starting point for our learning procedure is to view translation as a rotation in the high dimensional space of pixel intensity, right? So if you think about an image that's translated, translation doesn't change the contrast in the image. So the norm of the image in the sense of the sum of the squares of the intensity values at all the pixels should stay the same. There's some edge effect here, of course. So the pixels at the edge actually can change. So the norm does not stay exactly the same, but it should be a good approximation as long as we can ignore the edges. So we want to think of motion as this sort of trajectory that moves on a high dimensional sphere in the space of pixel intensity. And now the question is, how do we learn that rotation? And of course, translations are you can translate images by different amounts, right? So depending on the speed at which an object is moving, if you look at two consecutive frames in a video, the amount of motion is going to be different. So we want to somehow capture the essence of this translation, the direction in which it's going, and not worry so much about the amount. So mathematically, we represent that by an infinitesimal generator. And the idea is that if I can take something like a rotation, I can split it into very many small rotations. And the small rotation, if you think about it as a linear transformation, looks like the identity plus some small modification of the identity that implements the rotation. So you can think of it as this product of identity plus a generator A, or in the continuous form is the exponential some amount, some amount of rotation, some angle essentially times the generator. So as an example, if you have a rotation in 2 dimensions, this is the matrix that performs that rotation, and you can think of it as in this exponential form with a generator that's an antisymmetric matrix. And this is in general the case for rotations. The generators are antisymmetric matrices. So what we really want to learn are these generators, because those allow us to understand rotations or translations by any amount. So how do you do that? Well, whenever you have 2 vectors, you can imagine a rotation that turns one into the other. In this case, I show this big circle, and we can imagine a rotation that goes on this big circle from A1 to A2. But so that's one rotation, happens to be the shortest rotation, the rotation that requires the smallest angle to perform this transformation. But there are many other rotations depending on the plane in which we want to perform the rotation. So if we want to go along a different trajectory that's not a big circle, then we can get many different rotations that could do the same transformation. So that means that if we want to learn the particular transformation that converts frames at time t1 to frames at time t, we might need to average over many examples, because any pair of examples is not going to be able to identify the rotation we're talking about. In a more mathematical language, the sort of rotation we get in this case is a rank 2 matrix, but the sort of rotation that we care about if we're looking at global translation, for example, is a full rank, so rank equal to the total number of pixels in the image, which could be very big. Okay, so now in an actual natural environment, we can have several different kinds of transformations. In the simplest case, we can imagine translations in a vertical direction and translation in the horizontal direction. So how do we distinguish that in an unsupervised setting? And a simple approach that seems to work is to just use clustering. And we could use any sort of clustering. So we would cluster these rotations that we're inferring from each pair of frames. But we want to think of this as a possible biological implementation of the learning algorithm. So we're using something developed in Mitya Shuklovsky's lab for the past few years, which is called similarity matching. And the basic idea is that you want to find the outputs that best match the correlation structure of the input. So if you have two inputs that are very similar, like these red arrows here, you want them to be mapped by two outputs that are very similar. And two inputs are very dissimilar, like the orange ones, should be mapped to outputs that are very dissimilar, like the orange ones on the right. So in our case, the x's, the inputs, are these basic rotations that we learn from every pair of frames. And it turns out, and I don't have time to go over this in detail, but it turns out that these need to be anti-symmetrized outer products. So I take the frame at time t+1 and the frame at time t. I make a matrix by taking the outer product of those two vectors, and then I subtract the transpose to get an anti-symmetric matrix, because that's what rotation generators are. They are anti-symmetric. So this objective function here doesn't look biologically plausible at all. I'm summing over all pairs of samples, but it turns out that we can actually write it as a very simple biological circuit that has Hebbian plasticity at these two sets of synapses. And this circuit itself on its own just does PCA, but it also turns out, and I don't have time to go over this in detail, but it turns out that if we impose the constraint that the outputs are non-negative, this circuit is going to perform clustering instead. So now we just can calculate the outer products from consecutive frames, feed them through this circuit, and out come an output that's essentially, the output here is essentially just a projection of the inputs with the matrix W, which is given by the weights in the circuit. So those weights are going to be our detectors, our motion detectors. So we're going to look at a very simple example in one dimension because it's easier to understand. So we're going to start with patterns that are essentially just plateaus like this of different lengths. So these are 6 different patterns. You see the pattern at time t and at t+1. So these are just plateaus that move by an amount between 0 and 3 positions in one time step. And then we're going to apply a sort of a filtering, a sort of centered surround filter to obtain these wiggles that are moving from one frame to another. And these are the frames that we're going to use for learning. After we run this with about 100,000 samples, these are 6 of the generators that were learned. So the thing that we notice here, remember that W's are the connections So these are the connections in the networks. And the things to notice is that, first of all, these are local. They respond only to a small portion of the image. And they're also local in the sense that you only need connections between nearby pixels in the image, right? So all the non-zero entries are localized in a small space here. And I'm sort of running out of time, but this looks like sort of classical models of motion generators. And to make this a little bit more intuitive, we can just make a plot. So for each of the filters, I made a pattern that has a peak kind of around the center of the generator and another peak somewhere where the generator is almost 0. And then I can apply the generator to these patterns, and I see that indeed it moves one of the peaks in one direction or the other, and the other peak stays about fixed. So this shows that what I've done here is I've learned generators that motion generators that apply locally. And yeah, I just said all of this. So I'm going to leave this here, and thank you for your attention.
All right, thank you so much, TB. I actually have a very naive question about how you would take into account like jitters in the motion. Like if let's say there is, like essentially how the retina performs the saccade correction or something like that. Do you have a similar type of correction that you apply where you're canceling out the effect of jitters in the movement so that you're not actually applying some type of an actual transformation term to it?
Right. No, we haven't yet thought about this in detail, and it's sort of two different ways of looking at it, right? So it might, in some sense, most of the motion observed by the retina might come from this sort of self-motion, from the eye moving. And it might be that that's the sort of motion we should be focusing on rather than the actual motion of objects in the world. So I guess I should say that in this sort of artificial example, we haven't specified where the motion comes from. This could be just from the eye moving rather than from the objects themselves moving. But yeah, we haven't thought in much detail about how those two things interact.
Right. And another, I guess, very naive question is, so in this, your matrix, so all these different inputs are essentially different positions of the moving object, right? Like, so you're taking all of those into account, and then, or essentially you're applying, so all of these different inputs are different objects moving, right? As opposed to different movements of the same object.
I guess in some sense that's right, because I'm randomizing the sizes of the objects in each sample too. Yeah, I don't think that makes a lot of difference, at least as long as we have some variability in the size of the object. So one thing that's sort of striking here is that objects always come in pairs, right? There's an edge and another edge where the object ends, but the filters that we find don't necessarily come in pairs, right? So some do have both sides, but some somehow focus on only one side. And I think if you had objects that were always the same size, then you might get filters that always have two kind of peaks. But I think as long as you have some variability in size, it's not going to happen.
Right. All right. Well, thank you so much, TV. That was really great. We're going to move on to our next and final speaker, Dr. Mina Zareh. She is an assistant professor of physics at the Institute for Advanced Studies in Basic Science in Iran. And she's going to talk to us about the synchronization of adaptive networks of Sakaguchi-Kuramoto oscillators.
Hello, everybody. I'm going to speak about the synchronization of adaptive networks of Sakaguchi-Kuramoto oscillators. The motivation behind this work is that if we consider the cortex, we can, in the resting state, we can see that we can see that there are lots of the nodes with high frequencies, the yellow ones, and a few nodes with the low frequency, the blue ones. And in addition, frequencies are assumed to be a function of the anatomical node strengths. I mean that these blue nodes with low frequencies have a high number of connections, and the yellow nodes with high frequencies have low number of connections. This is from higher to lower connectivities. For example, we can see it better in the schematic graph. it looks like a star, that the hub is the low frequency nodes with a lot of connections, and the high frequency nodes have a low number of connections. The question is that, can adaptation lead to these structural differences between the high and the low frequency nodes? So we are going to think about this during this seminar. Here I use the synchronization dynamics. You know this. For example, here we see the synchronized movement of the dancers. If we have a network of phase oscillators, then we couple them together, we can see the time evolution of the phase of each oscillator according to the chromato model. Here, theta dot is the time evolution of the I oscillator. Omega is its natural frequency. Lambda is the coupling strength. And R is the adjacency matrix that the structure of network is hidden inside. So here we have the interaction term. If we look at this equation is time reversal symmetric. I mean that if I change time to minus time, nothing will change. But if I add a mismatch parameter to the equation, which is called the Sakaguchi-Kuromoto model, mismatch parameter breaks time reversal symmetry. We are going to see that this parameter, how affects the dynamics and structural changes of the network during the adaptation. At first, I need a measure to quantify the level of synchrony. I use the chromato order parameter, which is defined like this, and it changes from 0 to 1. When it close to 1, we are in the synchronous state. I mean, the phase of oscillators are close together. And when it is close to 0, we are in the asynchronous state. I mean that the phase of oscillators scattered along the unit circle. If you have a network of phase oscillators that are evolved according to the chromatodynamics. At first, they are not synchronized, so the order parameter is close to 0, and after a while, their synchronization increases to the certain level. The synchronization value at the infinite time or a stationary time is called a stationary order parameter. When here I speak about the order parameter, I mean the stationary order parameter. But suppose that we have a network that with phase oscillators connected to each other with the chromato model. The nodes are different and everything. But the reality is that the structure of the network is not really fixed in reality, but it changes during the dynamics. There are two well-known adaptation rules, Hebb rules and anti-Hebb rules, that both of them are important for the function of the brain in the memory, in the learning, and vice versa. Hebb rules, we can say that if the connection between two nodes that are often synchronized during the dynamics will be strengthened after a while, and the connection between two nodes that are often not synchronized during the dynamics will be weakened after a while. The anti-Hebb rule is opposite, and the Hebb and anti-Hebb rules, both of them lead to different structures. Here we use the rewiring two different rewiring processes to model Hebbian and anti-Hebbian actually learning. Suppose that in a time during the dynamics, two nodes are connected and they are asynchronous. Their phases are not close. We rewire the links to another random node. So after a while, when we do this several times during the dynamics, then the links between the asynchronous nodes will be weakened, and then the links between the synchronous nodes will be strengthened. So this is the Hebbian rewiring. in anti-Hebbian rewiring, we did reversal things. So if two nodes are connected and they are during the dynamics almost synchronized, we rewire the links to another random nodes. So during the dynamics, the links between the synchronous nodes will be weakened. So we have the anti-Hebbian rule, we have the Hebbian rules, and these two change the structure networks in different ways. Here are the results. If we have in fact, the chromato model without the mismatch parameter and anti-Hebbian rewiring, suppose that the initial structure of the network is homogeneous. I mean that we have a uniform frequency for each node, but for each node, the number of links or the degree of each node is also similar. We run the chromato model on these networks for a certain value of the coupling constant, and we see that during the time the order parameter is low, so it's not synchronized. We started to do the rewiring according to anti-Hebbian rules, and we see that when we do the different rewirings, the synchronization increases, and after a while, it reaches to the stationary high level of global synchronization. So the anti-Hebbian rewiring leads us to the global synchronization and how it changes the structures. If we see the structure is not homogeneous, it's inhomogeneous, and here the size of the nodes shows the number of links that connected to that nodes. So we see that there are some nodes with a high number of links, which we call the hubs. And the color shows the actually the frequency of the nodes. So gray is the mean frequency of the nodes. Red is high frequency of the nodes, and blue is low frequency of the nodes. So the nodes where the frequency are far from the average, actually they are hub. They take a lot of links. And we can see this here better. Here is the degree of the nodes that I said, the number of links that connected to each node. And here are this value, I mean that the natural frequency of each node minus the average frequency. So when we say 0, it means that the node has the average frequency. When we say -2, the node has a lower frequency than average, and 2 is higher frequency than average. We see that there is a correlation between the degree of the nodes and the absolute value of these values, or we say that the natural frequencies. But if you remember the motivation part, in the resting state, we see that the nodes with low frequency has a high number of links, but the nodes with high frequencies has low number of links. So this adaptation doesn't lead out to that correlation that we mentioned in the motivation. But if we add a mismatch parameter and do again the anti-Hebbian learning, we see again that anti-Hebbian learning leads to the global synchronization, but less than when the alpha is 0. And if we plot the similar plots like before, the degree of each node versus the each frequencies. So 0 is the average frequencies. Here we have nodes with low frequencies. Here we have nodes with high frequencies. When alpha is 0, we have symmetric around the 0. But when we add the mismatch parameter, that can be really a small amount of the delay in the network, and the time reversal of the equation will be broken, then the symmetry around the mean also will be broken. Now we have the nodes with low frequencies have a higher actually degree than the nodes with high frequency. It's similar to the things that we said in the motivation. And this minimum will be changed. We can find it analytically. I don't speak about this now. But how about the Hebbian rewiring? If we do Hebbian rewiring, we don't have a structure with global synchronization, but we reach to the clustered structures that in each cluster, the nodes are synchronized, but the clusters are not synchronized to each other. So if we plot the adjacency metrics for different mismatch parameters, we will see these clusters. So here the colors shows the probability that two nodes are connected. So yellow ones that they are almost connected together. We see and the nodes are ordered from the low frequency to the high frequency. So the node 0 have the lowest frequency, the node 100 highest frequency, and the node 50 almost the mid frequency. We can see that when we have zero mismatch parameter, I mean that the chromato equation we have, and we have time reversal symmetric, and evolve the network with these dynamics, we will reach to a structure that it is symmetric around the mean frequency. So we have a large cluster around the mean frequency and small clusters around these large clusters. But when we add these mismatch parameter, the symmetry around the mean frequency will be broken, and the nodes with low frequencies will have a higher number of links than the nodes with high frequencies. So if we want to have a network that we said, like the things that we said in the motivation, that the nodes with high and low frequencies have different structures, we need some mismatch parameters or some delay in our dynamics. The results is that the anti-Hebbian dynamics leads to the global synchronization with frequency degree correlation correlated structures. The Hebbian dynamics leads to the clustered structures with local synchronized clusters. And the mismatch parameters actually causes the structure of the evolved networks have asymmetry respect to the mean natural of frequencies, as we see something like that in nature. These are the collaborators of these works, and thank you for your attention.
Thank you so much, Meena. That was really great. We had a question in the chat, but I think you've already answered that, which was about the definition of adaptation. And I also actually had a very similar question, because to me, adaptation is, the way I think of adaptation is more on, the network adapts, and so it no longer fires anymore. And so you would expect there to be either lesser degree of connectivity or some other type of anti-connectivity in the sense. So I was wondering if you could elaborate a little bit more on the actual definition that you used for adaptation.
In the adaptation, actually, the structure is changed during the dynamics. So we have always different states for each node during the dynamics. And it's a little difference between the Hebbian that we define here and the Hebbian that we defined like STDP, because they have that phase lag in the time. But in each instant of the time, actually, according to the state of the nodes at that time, we will have this adaptation until that we reach to the stationary point. I don't know if I answered your question or not.
Yeah. I had a related question regarding how stochastic these processes are in terms of, like, if you shuffled all the nodes, let's say, and you had a different starting structure, then how would that influence your
It's really robust. Yes, it's really robust. So we did it. Actually, the results are for the average of our different initial conditions and the different networks, and the results are qualitatively robust.
That's really cool. All right, yeah. Thank you so much.
Thank you.
I don't think we have any more questions, and I think we are right on time for the session to end. So I would like to thank all of you for the very wonderful talks. Thank you so much. Thank you everyone for your attention.
Okay, guys, we're live. I'm going behind the scenes. Good luck.
Okay, my clock says that we're right on the hour, so we can go ahead and get started. Welcome everybody to the final day of NeuroMatch 3.0. I'm Emily Airy-Jones. I'm a postdoc in the Giacomo Lab at Stanford. I'm going to be your front-end host today. We have Nicola at the back of the house, who's got his camera just turned off. We have an amazing lineup of speakers today. Today's session is about odor representations, first in the hippocampus, then in lateral entorhinal cortex, and lastly in the olfactory cortex. We want to encourage everybody to post questions to the Q&A. You can upvote questions that you think are interesting. We only have 3 minutes to talk about questions between sessions, but we encourage all the speakers to ask and answer questions in the chat afterwards. And everything here is public, so please share it on Twitter with the hashtag NMC3. Our first speaker today is Yanis Taxidis. I hope I'm not butchering that too badly. A project scientist in the Golshani lab at UCLA. Today he's going to be presenting his findings on odor and time and how they're encoded in CA1 spiking sequences and how these fields adapt over days. My own postdoc project is on sequences, so I'm really excited to hear about how sequences encode odor and time beyond our usual understanding of just spatial sequences in the hippocampus. So please take it away.
Sorry, I forgot that I was muted. Let's go again. Okay, so my talk today is all about the overarching question of my talk is remembering what happened to us and in what order, what happened when. So at the end of the day, we can easily recollect all the events, the major events that took place during that day, and we can put them in the right order. This thing happened first, about an hour later, I did this, and so on and so on. How does the brain do that? Well, we know that the hippocampus is necessary for us to perform that basic function. We know that because, for example, if you're asked to walk around a campus and asked to do a bunch of tasks like pick up a coin, drink some water from a fountain, buy an item from a cafeteria, and so on, once you return back to the base and you're asked what happened, you can easily list the tasks that you took, but also you can put them in the correct order. Shown here in black is healthy individuals. I can nicely correlate the actual order of things, the actual order of events with their recollection. If you have a hippocampal damage and you do the exact same thing, not only you have a problem remembering what you did once you return back to the base, but you also have a really hard time putting the tasks that you participated in the correct order. So shown here in white, it's completely uncorrelated what you report as first, second, third with what is reality. So we know that the hippocampus is necessary for creating a model of our experiences and the time it took between them and putting them in the right order. So the question is, how does the brain, how does the hippocampus create a model of that? How does it represent the various events that are taking place and keeping track of the time interval in between them so that you can stack them in the right order? Let's simplify the question a little bit. So when you're talking about a mouse or an animal, an experimental animal, you might say, okay, I have a sequence of sensory stimuli, like a very basic odor. It was followed by, let's say, a visual stimulus, then maybe another odor came on and so on, with some time intervals in between. And the question is, how does the hippocampus create a representation of all of that so that then it can be stored as memory and be retrieved later? Well, we know that the hippocampus generates representations that come more or less in two flavors, let's say. One is sensory representations, representations of the external world, sensory cues that are coming into the brain. So for example, if a mouse needs to pay attention to a sound that is followed by an odor, then the hippocampus generates a sequence of neurons that are firing one after the other during the sound presentation, taken over by a different sequence that fires during the odor presentation, and so on. So we have cells or sequences that cover these sensory cues, direct sensory inputs coming in. For the time intervals, we also know that the hippocampus generates, again, representations, this time internal, we call them internal representations. And those can be representations of place, the very famous place cells, or representations of time. We have time cells as well. For example, if a rat needs to remember a cue while it's trapped in a part of the track here for a few seconds, the hippocampus generates sequences of neurons that fire one after the other, like the one shown here. Essentially, for us, the decoder, keeping track of time elapsed into this entrapment here. So basically, these neurons, one after the other, tile the interval in between the events. So put these sort of two kinds of representations together, and you can say, I have the hippocampus generates these sequences encoding the actual sensory input. So let's call these sensory cells. And for the intervals in between, it generates sequences of time cells. So that if later on if I knew exactly what each cell here represents, I could just look at this sequence and know that, oh, there was an odor that came on here, and then 1, 2, 3, there was 5 seconds here, and then a visual cue came on, and so on and so on. So I created a model representation of the experiences and the temporal intervals in between. Now, people usually study either these sensory representations or time cells or place cells separately. So my question was, putting these things together, does the hippocampus generate these external and internal representations using the same dynamics, or does it use different modalities for the two kinds of representations? So let's break this question down a little bit. So first, can sequences truly combine these representations of external cues and time intervals? To answer that, we needed a task that combines both. So we trained mice to perform an olfactory delayed non-match to sample task. The trial is initiated by an odor. It can be either odor A or odor B. And then 5 seconds later, again, an odor comes on. It can be A or B. The mice are trained to remember the first odor throughout these 5 seconds and compare it with the second one. If the two odors did not match, so it was an A-B or a B-A trial, they lick to get a reward from a lick port. If the two odors were the same, an AA or a BB trial, they reject, they don't lick because there's no reward. So in this case, we have an important sensory cue that the mouse needs to remember, and then an important temporal interval during which working memory is activated in order for the cue to be compared with the second cue. Okay, so if we want to, so then I talked about if the dynamics are the same for these two representations. So the question specifically is, how stable are these representations across days, or if we change something in the trial and the context of this trial, and so on. So if you want to talk about stability, you want to be able to track the neurons across days. So our mice are head fixed on a styrofoam ball, free to run whenever they want. They're under a 2-photon microscope. So I've removed the cortex overlaying the hippocampus, and I put a cover slip so I can perform 2-photon calcium imaging on the dorsal CA1 pyramidal layer. So if I do a nice surgery, I get thousands of cells here. In red, you can see interneurons. These are mice that express TD tomato in all their interneurons. So I can image the interneurons in the red PMT channel and then remove them from my analysis. Everything I'm going to show you is only from pyramidal cells, which are shown here in red. So as the mice are performing the task, we are recording nice videos like this and tracking the activity of these neurons. And finally, the last question is, so if these sequences really exist, do they emerge during learning of this temporal context and so on, or are they pre-existent sequences that are just getting assigned to these trials? And so to answer that, you don't need a task that will take months and months and months for mice to learn, because it's really impossible to track these neurons nicely for that long time. So luckily for us, the mice can really learn this task and perform really well within 5 days or so. Okay, so what do we see? So let's split the trials in the ones that start with odor A, shown here in yellow, versus the ones that start with odor B in green. So what we find is we have a sequence of neurons that are tiling starting from the presentation of the odor and tiling the entire delay. And this sequence more or less repeats itself if the second odor is the same as the first one. So you have a bunch of cells that are representing the actual delivery of the odor. And these are followed by a sequence of neurons, the so-called time cells, that are tiling the entire delay. And notice that these neurons are quiet when the trial was initiated by the opposite odor. So if you just focus on a neuron that's sitting there, these are average firing rates. Now, if you look at a neuron that's here, basically you look at all the trials, and here I've stacked the trials based on the sequence of odors. So this neuron likes to fire when order A is presented, and it doesn't really care if it's the first order in the trial or the second order. It fires right at the presentation of order A. So a neuron down here, on the other hand, fires about 2 seconds into the delay, but only after order A was presented, not in trials that were initiated with order B, where the cell is more or less quiet. For the case of the order B trials, you have a different sequence, another group of neurons that are doing the exact same thing, but just for the opposite order. So an order cell up there will be firing only when order B is presented in green, and a late time cell here will be firing towards the end of the delay, but only after order B was presented, not after order A. Okay, so that kind of answers the first question. Can sequences combine representations of external cues and time intervals? Yes, I've shown you two different cue-specific sequences that combine representation of both the stimulus and the time interval. Oh, sorry. So the second question is, how stable are they across days or changes to a temporal context? Let's start with the second part, the temporal context. So the easiest way to change the context is to just extend the delay, right? From 5 seconds, we just extend it to 10 seconds. Mice keep performing the task, but they are just suddenly introduced to a 10-second delay and they keep going. What happens to the cells? So this is an example of an odor cell. These are all the trials of 5-second delay, and this is the firing rate of the cell during those trials initiated by the odor that the cell encodes. This is an early time cell firing just at the onset of the delay, and this is a late time cell firing towards the end of the delay. Now, what happens when we suddenly introduce the 10-second trials instead of 5 seconds? The odor cell keeps firing here. The firing rate drops a little bit, but it retains a significant field very nicely. The early time cell goes completely quiet. Late time cell becomes completely disorganized here and loses its field. So if you look at the sequence as a whole, you have a nice sequence for the 5-second trials. The exact same neurons stacked the exact same way for the 10-second trials. The time sequence here is gone, even though the odor cells are nicely stable. Okay, so you have a remapping of this sequence. I should say here that these trials are tiled by their own sequence. It's just a new set of neurons. The exact same thing happens across days. So we track the same field of view for multiple days and we follow the neurons for the same neurons across days. And what do we see? Take any day, day X, and you find a nice sequence of cells. And if you look at the same neurons the next day or the day after, again, we stack here the neurons the exact same way. The firing rates here are pretty much conserved. So the odor cells remain odor cells with a nice significant field firing during the preferred order, whereas the nice sequence of time cells is already jumbled up the next day and pretty much gone 2 days later. Again, each day has its own sequence that looks pretty much like this. It's just composed of new time cells, even though the odor cells are more or less the same. So stable order representations across days or across changes, but flexible temporal representations. Finally, how do these sequences emerge during learning, if they do? So again, this is the performance of the mice as they're learning the task, and we image them as they're learning. This is the sequence I get from a bunch of mice on first day of learning. So you have some odor cells, and here's some time cells. I separated them by a dashed line. And this is the exact same mice imaged, the same field of view for each one, imaged 6 days later when the mice are now experts in this task, and you get about, again, some more or less 150 odor cells, but way more time cells. In fact, if you just count how many odor cells you have every day from these mice, you have a pretty much flat line here. Whereas if you count how many time cells you have, it nicely correlates with, it nicely increases across days. So basically the number of time cells is very nicely correlated with performance, whereas the number of odor cells is not at all. It remains fixed. You might say that this has to do nothing with learning, and it's all because we trigger these odor cells over and over and over again because we present these odors for many days. It has nothing to do with learning. So to address that, we repeated the experiment with completely untrained mice that received the exact same trials, but they didn't know what to do with these trials. They were not trained to look for a reward and so on. On day 1 and day 6 from these mice, we get the exact same, more or less the same sequences. Same number of odor cells, number of time cells is completely flat. It does not, the number of time cells does not increase. So you need the active learning of the trial and the context to increase your time cells. So to conclude, time representations emerge or expand during learning of a temporal context. And just to put this all together, I asked, are external and internal representations governed by the same dynamics? And I hope I've convinced you that the answer is no. We get very stable representations, stable odor cells for fixed external cues, the odor coming in. Whereas for the variable temporal intervals in between these experiences, we have very flexible and dynamic and learning dependent representations. And I would argue that this combination of stability for stable things coming in from the external world and flexibility for variable temporal interval representation might be necessary for the hippocampus to be able to construct these maps of what happened when and turn them into memory. I would like to thank everybody in the lab that has helped more to some degree or other with this work, and of course Payman Golshani for his mentorship. This work was published in Neuron a few weeks ago, and thank you very much for your attention. I'm happy to take questions.
All right, thank you so much. We have maybe 1 minute for questions, and so while questions are coming in, oh, we have one already. Awesome. All right, so Kara, sorry if I'm mispronouncing that, Wiseman asks, I'm wondering why the time cells increased with training when the time between stimulus and requested response wasn't something that the mice had to learn, or did I misunderstand the task? The training was to respond based on differences between the stimulus, not to count time?
Exactly, that's a very fair point. So basically what I claim here is that the time cells are not really keeping track of time. The hippocampus is not required to count first second, second second, third second, fourth second, and so on. What it needs to do is just remember the first order, right, throughout the delay. So what these time cells do is they sort of hold this information because they're order specific, the sequences, they hold that information up until the second order comes on. And what's happening here is that as the mice are learning this task, they learn that the first order is important and I need to remember it, and they engage their working memory more and more. The hippocampus seems to be throwing more time cells into this task, building these sequences so that this information is carried over and up to the second order more and more efficiently. That's what I think is happening.
All right, thank you so much. So we're gonna move on to our next speaker, and if you guys have any more questions for Yanis, then please put them in the Q&A and he can respond to them in the chat. All right, our next speaker is Sebastian Bitzenhofer, who's a postdoc from the Isaacson Lab at University of California, San Diego. He has a recent preprint out from his thesis work on how transient exogenous prefrontal activity impairs neuronal maturation and cognitive function, which I will link in the chat. While in our last talk, we heard about odor representations in the hippocampus, now we're going to move one synapse upstream to the lateral entorhinal cortex.
Hey, thanks for the kind introduction. Let me quickly put on the laser pointer and then I think I'm ready to go. Okay, so animals rely on olfaction to find food, attract mates, and avoid predators. And this requires the identification of the same odor across a range of intensities, but also it requires discrimination of odor intensities themselves. And this is especially important during tasks like navigation, for example. When we look at the olfactory system, odor molecules activate the peripheral olfactory neurons that project to mitral cells in the olfactory bulb, the initial odor processing station in the central nervous system. And these mitral cells then in turn project to several areas, including the piriform cortex, the primary olfactory cortex, this large portion here. So a recent paper from Kevin Bowling and Franks shows that odor-evoked activity in the olfactory bulb comes in two phases, and this is known for a long time. And it's shown here at the mean firing rate of neurons recorded from the olfactory bulb. They show that there's an initial phase that is largely invariant for different odor intensities. And then there's a late phase that is changing more dramatically if they present different odor intensities, shown here from yellow to red. In contrast, the piriform cortex, so the primary olfactory cortex, only responds to this initial phase. And they show that recurrent connectivity in the piriform cortex suppresses the activity during the sustained phase. And we conclude from this that the piriform cortex represents odors invariant to intensity, allowing to identify the same odor across a large range of concentrations. So, but odor intensity is also important. So where is odor intensity processed? There are several other areas that receive input from the olfactory bulb. So we wondered, since odor intensity is especially important during navigation, if you, for example, want to find food or something, we wondered if the direct projections from the olfactory bulb to the lateral entorhinal cortex may transmit this information about odor intensity to the entorhinal hippocampal network. So to test this hypothesis, we performed extracellular recordings from awake head-fixed mice, simultaneously from the olfactory bulb, piriform cortex, and LEC, or from the olfactory bulb, LEC, and hippocampus. We used a pressure sensor to record the respiration of the mice and give a closed-loop odor stimulation triggered by the exhalation of the mice. So when we look at the PSDHs that we give from this exhalation-triggered odor stimulation, they look quite blurry in time, actually. So we realigned the trials to the first inhalation that is coming in after odor onset. And when we do that, we see that odor evoked activity is actually quite precisely timed for this example neurons from the lateral entorhinal cortex. So we looked at the average activity for all units in these areas. And as described before, we see this initial phase, and if we give odors shown here in blue, at about within the first 100 milliseconds after inhalation in the olfactory bulb, and then this late phase, the sustained phase that is coming later on from about 100 to 400 milliseconds. And again, we saw that the piriform cortex only responds to the initial phase. To our surprise, however, the initial phase of odor evoked activity also strongly dominated in the lateral entorhinal cortex, as well as in dentate gyrus and CA1 of the hippocampus. So we wondered, is the LEC actually important for the discrimination of odor intensities? We decided to test this using two alternative forced choice tasks in mice. We used get-to-agree mice to express gyrodopsin selectively in interneurons, which allows us to transiently silence the LEC. First, we trained mice to discriminate between two odors and reported choice by licking left for odor A or right for odor B. And similar to the electrophysiological recordings, odor representation was triggered by exhalation. To our surprise, simple odor discrimination was impaired when we silenced the LEC bilaterally, which is kind of unexpected because it was supposed to mainly depend on the piriform cortex. And when we looked at the timing of the lick responses of error trials and correct trials, we see that already at about 200 milliseconds, the mice are able to distinguish correct choice and perform above chance. So that indicates that this fast component that this initial fast component is important. And again, here this timing was aligned to the first inhalation after odor onset. Now, when we trained mice to discriminate intensities, they were also able to do the task, but again, their performance dropped to chance when we silenced the LEC. And again, they were able to discriminate the two intensities already about 200 milliseconds after inhalation. So sometimes they respond later. We don't force them here to respond fast, but that's when the correct trials and error trials basically are separated from each other. So this shows that the LEC is critical for the discrimination of odor intensities, and the timing suggests that the initial phase of odor evoked activity does not only contain information about odor identity, but also about odor intensity. So we decided to look at the local network in the LEC in more detail to understand what is going on. We focused on layer 2 of the LEC, which is the main input layer for olfactory information and the main output layer to the hippocampus. There are two different cell types in layer 2 of the LEC, fan cells and pyramidal cells. And fan cells express reelin and are located in layer 2A, which you can see here, whereas pyramidal cells express calbildin and are located a little bit further away from the surface in layer 2B. We identified a new driver line, Netrin G1, pre-mouse to selectively express target expression in fan cells. And expression in pyramidal cells can be selectively targeted with calbindin driver line. We used Netrin G1 mouse line to express TD tomato and fan cells of the LEC and looked at their projections to the hippocampus. And we found that fan cells project to the outer molecular layer of the dentate gyrus by the lateral perforant path. In contrast, when we use Calvin-Cree mice, we see that pyramidal cells directly project to the stratum maculosa moleculare of CA1. So fan cells project to the first station of the trisynaptic hippocampal loop, the dentate gyrus, whereas pyramidal cells project to CA1, the last station of the trisynaptic loop, where the two pathways actually converge again. We patched fan cells in LEC slices and filled them with biocytin to look at their anatomical and physiological properties. And while we found subtle differences in their physiology, actually the most dramatic difference was the anatomy. The fan cells show very distinct dendritic morphology than pyramidal cells and span a large area at the cortical surface where the direct projections from the olfactory bulb arrive. So we performed paired recordings of fan cells and pyramidal cells and optogenetically stimulated incoming axons from the olfactory bulb to compare the direct inputs of the two cell types. We found that fan cells receive much stronger input compared to pyramidal cells from the olfactory bulb. Minimal stimulation of single axonal fibers showed no difference between fan cells and pyramidal cells, indicating that there is no difference in synaptic strength that two cell types receive, but in the number of inputs. When we stimulated axons coming in from the piriform cortex, again, we found that fan cells receive stronger input than pyramidal cells. However, the difference is less dramatic than for the input from the olfactory bulb. So, but what does this mean for odor-evoked activity? To address this question, we performed extracellular recordings from the LEC in array cat-fixed mice, expressing chenorodopsin in fan cells or pyramidal cells, which allows us to identify them by phototagging. And we looked at odor-evoked activity of individual units, and each line here represents the mean activity of one unit to different odors. We saw that the initial phase of odor-evoked activity within the first 100 milliseconds, dominating the response in fan cells as well as pyramidal cells. This becomes even clearer when we look at the average odor-evoked activity for fan and pyramidal cells. So again, fan cells respond to the initial phase, not to the late phase, and the same is true for pyramidal cells. So when we looked at the precise timing of the initial phase, so this is an expansion of this part here, basically overlaid, we saw that fan cells respond before pyramidal cells, which is consistent with the strong direct input from the olfactory bulb. And it's interesting because they are basically activating the local inhibitory network, maybe suppressing further activity. So, but what about odor intensity? To test this, we presented odors at 4 different concentrations, shown here by different color situations from low concentration to high concentration. And again, we only look at this initial phase where we actually saw clear responses. And we see that fan cells increase their firing rate with increasing odor intensity already during this initial phase. Pyramidal cells also increased their firing rate, but additionally they responded faster with increasing odor intensity, indicating an additional temporal code. So peak times of the odor responses at different intensities are summarized here and show the faster response of pyramidal cells, which is also there, but way less dramatic in fan cells. And this results in a decreasing time difference of fan cell to pyramidal cell activity with increasing odor intensity. So that's interesting because for downstream area, a temporal code only makes sense if there's a reference signal, since it does not have the information when the inhalation has happened. So our data suggests that fan cells might provide this reference signal, and the timing of pyramidal cell activity relative to the fan cell activity can be used by a downstream area to read out odor intensity, which might happen in CA1 where the two pathways actually converge again. So to summarize, I showed you today that mice can rapidly discriminate odor intensity, and this requires the lateral entorhinal cortex, and especially the initial phase of odor response in the lateral entorhinal cortex. LEC fan cells receive more direct input from the olfactory bulb and respond before pyramidal cells. Fan and pyramidal cells increase their firing rate with odor intensity, and pyramidal cells additionally respond faster as odor intensity increases. And this timing difference between fan and pyramidal cells could encode for odor intensity. Yeah. This, I want to thank to the lab, everyone in the lab, and the funding, of course, and thanks for watching.
Thank you so much for that lovely talk. While we're waiting for questions to come in, we have a few minutes for questions. I had a question which was related to the fact that the fan cells and pyramidal cells project to different regions of the hippocampus. Do you have speculations as to what then would happen to those hippocampal representations, that you would get, say, fan cells that have more olfactory inputs coming in projecting to dentate versus pyramidal cells to CA1?
That's something that we want to look into next, basically what is going on in the hippocampus. So one idea would be that the fan cells that get a more direct input from the olfactory bulb, it has recently been shown that the odor representations in the olfactory bulb are very stable, whereas odor representations in the piriform cortex change over days quite a bit. So maybe the direct projections onto the fan cells and then their projections to the dentate cells also result in a more stable odor representation over days in the dentate gyrus, whereas the one in the CA1 might be more varying with time. But yeah, that's what we were planning to look into.
Kind of as a follow-up, have you thought about looking into whether dentate is more involved in odor discrimination, similarly to how it's more involved in, or hypothesized to be more involved in, pattern separation?
It's a good question. We didn't look into this yet, but it would be interesting to do for sure. Yeah.
Okay, well, I'm sure that we will have more questions come in from the audience, and please answer them. You can answer them right in the Q&A, as Yanis has been doing. Sure. And so we keep on time. Let's move on to our last speaker of the day. Our third speaker is Hong Luo, a PhD fellow from the Schmitz and Johannick labs at Charité Medical University Berlin. He's also an eLife ambassador, and he has a paper that he helped contribute to. I will link that in the chat. He will be presenting something a little different from our last two talks, odor processing, in the cortex that's supposed to be involved in odor processing, the olfactory cortex. So please take it away, Hong.
Okay, thank you very much for the kind introduction. So, sorry, just let me start my laser pointer. So I'm a PhD student from Johann Schmitz lab from Berlin, and today I'm going to tell you a little bit about how the odor is being processed in the olfactory cortex in the scenario of eating. So this is just a work in progress, so it's not a defined project properly. So you'll see a lot of stuff is still really preliminary. So let's think about a scenario now. It's dinner time now in Berlin, and you're hungry. And now you see this beautiful cake in front of you. You have such a desire that you want to eat it, like just right now, because you smell the chocolate flavor, and they give you such a desire. You want to eat it just now. So we know that the olfactory system, the smell of food, contributes a lot to appetite. And also reports say that patients with eating disorders, they usually also comes with the smell deficiency. So in this context, to understand how odor is being represented or how it's being processed during the context of eating become a really important question. And so far it's not really being answered. So my PhD project focused on how the smell of food is being represented in the piriform cortex and during the context of eating. And we did up another experiment. We can learn the animal to start eating really, really fast. So we call it binge eating. So we perform clustering imaging in the piriform cortex. It's in the bottom part of the brain. we use a prism related green lens to relay this, so we can contact this to a miniaturized microscope. So we can do 3D moving imaging in the peripheral cortex. We develop one of the self-paced feeding paradigm, so the animal is not necessarily deprived. We only remove the food 5 hours before the experiment, so they will be a little bit hungry to actually go here and start eating. And we have a lick spout. They can trigger them, and they will receive a flavored milk. In the context I'm going to tell later will be strawberry milk. So this is the video. I hope you can see it. We have a lick spout here. When the animal triggers the lick spout, we will pump 1 droplet of the strawberry milk here. And around the lick spout, this is a glass tube connect to a suction. So we basically remove the air, the smell around the lick spout, so the animal will only smell something when they poke their nose inside. And you can see this is a miniaturized microscope. So we can see the calcium imaging when animals are freely moving and do all the behaviors. So what we found in the first part of my study is that when we deliver different flavors, because we have like different spout combined together, so we can deliver either the milk or the water to the animal. And what we see here is that some peripheral neurons have different activities, like the upper two, they have less activity when the animal is drinking water and milk. And the other two here, they show more activity when they're drinking water or milk. And this is a design that between each trial, there is a delivery delay, so they can only activate the pump again after 4 seconds before after the last one. So there's we design in this way to prevent the contamination of signals. We can also to get a much nicer signal from each trial. But we also do something else, is that we remove this limitation so the animal can drink as fast as they want. What we found is that when they're drinking really fast, the same neuron color-coded here, they basically have almost no activity in the peripheral, as shown here. So we also control for the speed of leaking, and basically it's not really different from when they're drinking a little bit slower or drinking really, really fast. So during binge eating, somehow in the peripheral cortex, the neural activity is almost nothing there. So this is the video from the GCAM imaging. You can see now there's some spontaneous activity, and later on, we'll get to the moment when the animal start binging. Like now, you barely see any calcium activity anymore. So let's run off for our experiment again. This is how one section of experiment is being done. And what I show you so far is the zooming part of this first binging, the first drinking boot, and also the second binging boot. And you can see here from the calcium events, most of the neurons here are suppressed when they are drinking really fast. Quick calculation about the calcium events, you can see more than 90% of neuron has a last calcium event when they're binging. So we later on look a little bit more into how the peripheral neuron process the information from strawberry milk and also from water. You can see here as that we use a AURC. Basically, it's calculating the area on the RC curve, represents that when the value is above 0.5 means neurons are more active. Below 0.5, they are more suppressed. You can see when the neurons are receiving, when there's a milk being delivered, some neurons are more active, some neurons are more suppressed. If we use the same sorting order to plot against their activity when the water is being delivered, you don't see a cloud, a red cloud here, means that those neurons who are responding to milk are less likely to respond to water. We can do the same for waters. So there are some also water positive neurons here. You can see in the plot here, and do the same using this sorting order for the milk neuron. You don't see much of the responses here. So we pick up some neurons. Like here, we can find like around a little bit more than 100 neurons. They have a positive activity to milk, and also they don't have much activity when it's being delivered to water, and vice versa for the water milk, for the water neuron. So since this effect is quite dramatic, and we apply the machine learning algorithm, supervector machine, and also the random forest classifier, we can get pretty nice accuracy, like above the chance level, which is 0.5 here. And we also start to think about, okay, if this calcium activity we see so far from the slow eating and also binge eating? Can we also classify them? And the result is yes, and it's surprisingly high. So basically we got almost 98% accuracy from this thing, from the classifier. So from the calcium data, we can easily tell whether animal is now receiving water or receiving milk, or whether they are slowly drinking or they are binging. Okay, so let's go back and look at this graph again. We know that during binging, there's a global inhibition take place in the peripheral cortex. But where does this inhibition actually comes from? We don't know this yet, but we thought about, okay, it could be just locally there are interneurons participating in this whole behavior. So we start with a GCAM imaging in the puff-cree animal. What we found is that surprisingly, during binging, the puff interneuron are also suppressed. You can see there are actually a little bit more activity when they're drinking milk and water, like relatively slowly, but when they're drinking really fast, this activity also goes down. So does it implicate that there are other interneurons taking place? We don't know yet, but other papers from Isaac's lab and also from Anna-Maria Ospa, they have illustrated this inhibitory neuron circuits, and most likely some of the standing interneuron could be taking place here. They can inhibit both excitatory neurons and also the puff interneuron. So we also start to think about whether this is actually effect from neuromodulation, because like it's strawberry milk and mouse leptin. So this could be just a rewarding system is acting up now. So we start looking into some literature and found that there's a potential mechanism that in serotonin neurons, if we activate a serotonin neuron as shown here, they do the recording in the prefrontal cortex and activity goes rapidly, almost nothing here. And also when animals drinking sucrose or eating food, you have a much higher activity in the serotonin neuron. So we think this is the potential mechanism like behind the thing. We're now still designing experiments to verify this, whether the effect we're seeing now is actually due to serotonin. And this will be the future experiment. Quick summary about what we have done so far. When animals eating, there are several neurons in the peripheral get lights up, which means these are potentially represent the flavor of this food. And with this lighting up neurons, they actually contribute to the concept of flavor or appetite, most likely in the orbitofrontal cortex. We don't really know yet, so we want to also look into the future whether we see this circuit actually contribute to appetite in orbitofrontal cortex. So when animals are binge eating, like drinking a lot of strawberry milk here, we see almost no responses in the peripheral cortex. Where does this inhibition come from? Is it actually come from some of the standing interneuron, or is a modulation effect from serotonin? Or this is like a synergistic effect of both? We don't really know yet, but this will be the future direction. So come back to my favorite slide. It's about the conjugate. I want to thank my supervisors, Fridio Henning and Dima Schmitz on the Charité, for all the support. Also for adding other people from Schmitz Lab, supporting other people, Marianne, Guyana, and Andreas Klaus. And because also many people helping me, like not necessarily directly linked to this project, but they're all listed here, especially thanks to my partner, Chiche. And my PhD fellowship is funded by Einstein Center, partly from Ministry of Education from Taiwan. Starting from next month, we'll receive from extra funding from Wellcome Trust. Welcome to email me or contact me on Twitter with any questions you've got so far. Okay, that's it.
All right, thank you so much, Hong. It was a lovely talk. We already have a question from Pierre Oran, who asks, what would be the purpose of these activities for suppression of the circuit?
So this is definitely a big question so far that we haven't really figured out. We have two theories right now. And first one is that maybe the only neuron, that might be only like 1% of the neuron in the peripheral is actually activating when they are binge eating. And so maybe these are the only important ones that actually contribute to flavor or contribute to appetite. So this global suppression of a neuron, of the activity in the peripheral is actually denoising all the other things. So they only focus on one thing. And the other thing is that we're thinking maybe this is not a cause by serotonin effect, but actually a reason, because when you have only a few neurons actually activating in binge eating, you actually receive much less sensory input in the peripheral. So that's why you don't get the sensory satiety when you're eating. So you actually eat much more than you actually should. So these are like two things we haven't really find a way to how to disentangle it, and maybe in the end we could do some either pharmacology or optogenetic stuff to try to see if we can actually understand, is this like the reason or this is just a consequence. So we don't really know the idea so far.
Thank you. I actually had a quick question, mostly because you mentioned interneurons, and I love me some GABAergic interneurons. So you mentioned that the parvalbumin interneurons decrease their activity, and then you mentioned that you have two hypotheses, one about suppression by serotonin, the other by somatostatin. So are you hypothesizing that the circuit is that the somatostatin interneurons are suppressing the PV interneurons? And how does like PV interneuron suppression relate to the fact that all of the activity, presumably in the excitatory cells, goes down?
So like don't need to go back. So basically, it's already been found that serotonin, somatostatin interneuron, they have this effect of suppressing basically everything there, probably besides the VIP interneuron. But for the PV interneuron, for excitatory neuron, for other somatostatin interneuron, they also have this really strong projection to them. So they have a really strong inhibition there. And to link this to serotonin, our lab actually just published a paper now is in bioarchive still. It's in Adorno Codex, and they link this serotonin effect. They can activate actually somatostatin neuron via the 5H2A receptor and suppress the slow oscillation in Adorno Codex. So I think maybe this circuit is also preserved in the peripheral. So it could be like serotonin, there is like sudden serotonin rush when they're binging, and somatostatin interneuron is activated and suppress everything there in the peripheral. That's so far our working hypothesis.
Neat. We also have Camille posted a link to a paper that she suggested that you might want to look at in the chat if you want to take a look. That's about endocannabinoids regulating olfactory bulb activity.
Oh yeah, I think I know that one. That one is awesome.
Yeah, awesome. All right. Well, if anybody in the audience has any more questions, I have put everyone's Twitter handles in the chat, so please follow up with them. I want to thank all of the speakers one more time. These were lovely and fascinating talks, and thank everybody in the audience for your questions and your chats and your participation. I want to thank the backend, Nicola, one more time for posting lots of things to the chat. Thank you. And everybody for ending on time so that I can take a 2-minute break before my next session. And I will see you all in the next NeuroMed sessions. Bye-bye.
Yeah, thank you so much. Thank you for hosting.
It can be, I mean, anything related to their expertise. And in the question on us, if you would like to ask your question on screen, please put on screen in your question and we will invite you to join. You can ask and discuss the topic you would like to discuss it with our speakers. We will start with our first speaker, Melanie Tobin. She is a postdoc in Maria Gaffen's lab of auditory coding at the University of Pennsylvania. She did her PhD in Paris on the biophysics of auditory hair cells with Dr. Pascal Martin, and now working on the role of interneurons in neural networks in auditory cortex. Her talk title is Role of Interneurons in Population's Neural Coding and Network Properties in Auditory Cortex. Stage is yours, Melanie.
Hi, thank you for the introduction. Yes, so I'm going to present my ongoing work on the role of interneurons in neural networks. Wait, sorry, when? Okay, good. Sorry about that. So when a sound arrives in our ears, it is transformed into an electrical signal that then is transmitted along the auditory pathway up to the auditory cortex. Within each region, there is a neural network composed of excitatory and inhibitory cells that work to process the sound signal. So within a network, the excitatory neurons receive the sound input and transmit the processed signal to other areas, while the inhibitory neurons help shape the response of the excitatory neurons. And so my question is, how do these interneurons help shape the response of a network to sound and help in processing. So there are many subtypes of interneurons actually in neural networks, and I'm focusing here on the three main subtypes of interneurons, namely the PV, SOM, and VIP interneurons. So the PV interneurons provide inhibition that is thought to have a role in maintaining the excitatory inhibitory balance, whereas the SOM and VIP neurons provide more specific top-down inhibition that helps to differentially modulate the network's activity. So how can we, if you look at this connectivity circuit, it is pretty complicated and intricate. So how can we figure out how one type of interneuron influences the other cells? Well, what we can do to figure that out is to look at the response function of the cell. So like the input-output function that has this sigmoidal shape here. And so if the cell input is the synaptic current into the cell and the cell output is the firing rate, we can measure this curve with and without activating particular subtypes of interneurons to see how the interneurons modify this response function, and they could have an effect on any number of these parameters or a combination of these parameters. So I set out to do this experimentally using the sound amplitude to modulate the cell input into the cell, into the network, using noise trains and 7 different amplitudes within 0 to 90 decibels, and then optogenetic stimulus to activate specific interneuron subtypes. And I've concentrated specifically on the VIP and SOM interneurons with by shining laser at 3 different amplitudes within 0 to 70% of the maximum laser power. And so then what I do is that I combine this auditory and optogenetic stimulus and measure the response of a network to these different combinations. And in practice, I inject the two viruses in the auditory cortex of mice. One is a calcium sensor that allows me to image the activity of hundreds of neurons using 2-photon imaging. And the second one is a flexed opsin that expresses specifically in VIP or SOM interneurons to activate them. And there is a red tag which allows me to then kind of identify these interneurons within the neural population. So when I activate the VIP interneurons within the network, let me start first by showing an example trace of a sound responsive neuron. So here this black trace is the change in response of a cell when there is no VIP activation, no laser, and we see that the cell responds to sound. And when the VIPs are activated, the sound response increases, and here you can see that it increases at all the sound amplitudes. And this may sound a bit counterintuitive because we're increasing inhibition and the response increases. Well, if we look at the connectivity circuit, we see that the VIP interneurons inhibit the SOM interneurons, which in turn inhibit the excitatory neurons. So activating the VIPs actually relieves the excitatory cells from the inhibition provided by the SOM interneurons. Now, if we look at the whole population of cells, we can classify them. So here in this yellow shaded area or this green shaded area, depending on whether they increase or decrease when there is sound. They may also not increase, not respond to sound. And then this red and blue shaded areas are similarly cells that increase the response or decrease the response when the VIP interneurons are activated. And what we can see is that for the vast majority of these cells, the response is increased. So here again, the black line represents when there is no VIP activation, and the pink lines are for the medium and high activation of VIPs. The response increases when the VIP interneurons are increased. So VIP activation leads overall to an increase in sound in the response to sound. What about the SOM when the interneurons are activated? Well, here again, I'm presenting an example trace of a cell that responds to sound. And when the SOM interneurons are activated, this sound response decreases and is even abolished here at the highest laser power. And this is actually more logical with how we think inhibition works, and we can see that the SOM neurons indeed project directly onto the excitatory neurons. If we look at how the whole population of response, how whole population of neurons responds, we can again classify them, regroup them depending on how they, whether they activated or suppressed by sound and or laser. And we see that the vast majority here, like all of these cells, see their responses decreased when SOM interneurons are activated. However, there is a small subset of cells which show an increase in their response when the SOM interneurons are activated. And so this is a bit hard to understand with the single cell analysis, this kind of like opposite effects on sound responsive cells. And although this connectivity circuit is helpful, what it hides is that there's not just 4 cells in auditory cortex, but a whole network of cells intricately connected to one another. And so this effect might arise from processing that happens at the network scale. So I look then at looking right now at what happens at the network level, and I'm using this tool called non-negative matrix factorization in NMF. And what non-negative matrix factorization allows is to basically kind of like decompose a network into sub-networks of cells working together. So I use this tool to find a smaller number of subgraphs and then select the subgraph which has the strongest expression during the stimulus presentation, and then look at the weights of each edge within the subgraph and average according to how the activity within to the sound and laser stimulus, the same that I described before. And so that way for each recording and each laser power, I can come up with this matrix of the mean weights for the strongest subgraph. So let me start with how VIP activation changes the network of the strongest subgraph. So here is the matrix for the mean weights across these different cell groups. For here on the left, there is no laser, and then medium laser, and here on the right, high laser. And the first thing that you can see is that the effect of VIP activation leads to this image basically showing a stronger, it kind of like enhances the contrast, the bright spots become even brighter. And so what that means is that VIP activation strengthens the subgraph that is already there. And if we look at the cell belonging, like which cell groups are within the subgraph, we can see that it's the sound responsive cells, the VIP cells here, but also the edges between these two groups. And then if we look at how the expression of the subgraph varies over time when the stimulus is presented, here the thick lines represent the average expression, but the thin lines represent the response to the different sound amplitudes from 0 decibel in the light gray to 90 decibels in black. And we can see that this mean expression is modulated by sound. With the SOM, when the SOM interneurons are activated, we see a different picture forming where the effect of SOM activation is not that the bright pixels become brighter, rather they become dimmer and new pixels become brighter. So what this means is that a new subgraph emerges when the SOM interneurons are activated, and the subgraph contains the SOM interneurons and all the edges to the sound activated cell groups and the sound suppressed cell groups. And if we look at the mean expression, this mean expression is not modulated by sound as much. So SOM activation leads to the emergence of a new subgraph, which is very much related to the laser activation. So here to summarize quickly.
Melanie, you have 20 seconds to finish.
Yes, so this is just like a very summary graphical summary where like the VIP activation leads to an overall enhancement of responses and strengthens the sound responsive subgraph, whereas SOM interneurons decrease most of the responses of the cells and lead to the emergence of a new subgraph. And finally, I would like to thank the whole Giffen Lab team and also my collaborators from the Bassett Lab, Danny Bassett and Jenny Slicer. Thank you.
Thank you, Melanie. This is very beautiful results, but unfortunately, we don't have any time for questions online. But if you have any questions, please write them in the question and answer panel. I'm pretty sure Eleni will be happy to reply them offline. Now we will move to our next speaker. And our next speaker is Badr. He did his PhD with Mike Deves at UC Berkeley to place theoretical bounds on the amount of information a neural network can represent given measured constraints on neural firing rates and correlations. He was formerly a professor of neurophysiology at Fordham University in New York and has just started a position at Pitt's Department of Neuroscience. He's interested in using computational and theoretical methods to understand how network dynamics produce perception and behavior with a focus on the auditory system. His talk is titled Distinct Synaptic Plasticity Mechanism Determine the Diversity of Cortical Neuronal Responses During Behavior. Stage is yours, Badr.
Thank you, Horiye. I should correct just one small thing because my collaborators will get very upset if I don't. So I was a professor of neurophysics. If my collaborators will not refuse to let me anywhere near an experimental rig for everybody's safety. So yeah, I'm purely a theory guy. All right, so I wanted to thank the organizers. Thank you, Horiye. Thank you for, I'm really excited for this talk format, and I'm excited to hear what kinds of questions people have, assuming I can get this down to somewhere approximating 5 minutes. I'm going to talk to you about a new project growing out of some older work, which uses a novel recurrent network model to understand the diversity of cortical responses observed in vivo. And let me just move this right here. Okay, great. Sorry about that. So it's well known that in awake behaving animals, many cells in cortical regions relevant to the task are not classically responsive in that they show no obvious firing rate modulation in response to task parameters. So here I'm showing you an example of what I mean by a sort of typical classically responsive cell. You can pretend this is an auditory cortex. Stimulus is played. You see a burst of evoked activity, and then it goes back to baseline. A non-responsive cell would look something more like this. This is the average firing rate over the course of the trial. And you see there's no obvious firing rate modulation. If you looked at it across trials or had some way of sort of averaging within trials, you'd expect to see something similar. So my collaborators and I are interested in asking a sort of simple question that was famously posed by Bruno Olshausen and David Field in 2005, which is, what could these nominally non-responsive cells be doing in the context of a task? So I just want to be very clear up front that I'm not saying they couldn't be responsive in some other context. I'm not saying there isn't a stimulus out there that wouldn't evoke them or make them suppressed, or there's other task parameters that you could lock to that would make them appear responsive. What I'm asking is could they be doing anything in the context of this task where there's no obvious task parameters where they appear to have some kind of evoked or suppressed activity? This is a follow-up to a previous study which was recently published in 2019, which was examining these cells. And in that study, we started by recording from either auditory or frontal cortex during a go, no-go frequency recognition task, where animals were trained to respond to a target tone for food reward and withhold from responding to non-target tones. So the relevance of auditory cortex to a task like this is probably obvious, but FR2 might be a little bit more mysterious to the audience. So FR2 actually goes by many, many, many names depending on what type of system you're interested in. They actually all overlap if you look at the atlas. And it's been implicated in sensory motor decision making, and it has reciprocal connections to auditory cortex. And using Musimon activation, my collaborators show that both regions are necessary for this task. So I'm just very briefly going to summarize what we showed in that study, since it's going to be the jumping off point for what I'm going to present here. So there's a couple of major findings from that paper. So the first one is that the majority of neurons in auditory cortex and in frontal cortex are nominally non-responsive to stimulus or behavioral choice. So about 55% in auditory cortex and 60% in frontal cortex. And this is just an example of one cell. This is the PSTHs of one cell where, you know, I think hopefully you can agree, aside from some statistical fluctuations, there's no obvious task-related modulation. Importantly, we showed that the ISI distribution of both responsive and nominally non-responsive neurons contains multiplexed information about stimulus and choice in both regions. And I don't say this here, but I should emphasize on single trials. So if you look at the spikes on a single trial, you can actually pull out just as much information from the non-responsive cells as you can from the responsive cells about the stimulus that was played and the animal's upcoming behavioral choice. And as much as I think we probably would have hoped that you would expect to see more stimulus information in AC and more choice information in FR2, that was certainly not the case. We actually found that FR2 contains more information about task-relevant stimulus category, and that's key. We're talking about the stimulus category, target or non-target, not the specific stimulus identity. And we also found that the one thing that distinguished non-responsive cells from responsive cells in terms of their decoding performance was that nominally non-responsive neurons were better predictors of behavioral errors than responsive cells. So this is the first sort of suggestion that we had that they may be playing a distinct functional role from responsive cells in the network. So I'm happy to answer more questions about this study during the discussion, but that's all we'll need to talk about what we're interested in next. So I've shown you that there are all these neurons that appear to be non-responsive. Where do these responses come from? They seem like they're important or potentially important, but how do we test this? And my collaborators are very interested in trying to get at this experimentally, but it's an extremely challenging task, which is to not say that it's impossible, but it's a really, really hard experiment. And once you get in there, you don't want to go in blind, so to speak. You want to have a clear sense of what hypotheses you might have about the different causes for non-responsive activity and the different roles that non-responsive activity may play. So for instance, if you're doing an activation experiment, you can target it at the points during the trial where you may expect there to be the maximal effect to differentiate these two subclasses. So we're going to start with a modeling study that tries to get at these two questions. What are the functional contributions of nominally non-responsive cells to behavior? What architectures and mechanisms produce these response profiles? So I'm going to start by presenting the model and just show you a few preliminary results and make way for your questions, hopefully. So what do we need for our model? We needed 3 components. So we needed a spiking recurrent neural network model, since we found that spike timings are important for our decoding algorithm to pull information out of these cells that don't modulate their firing rates. We needed to perform a task that looked similar to what we were asking the animals to do. And we included spike timing dependent plasticity as a mechanism for shaping the synapses of the recurrent neural units, because our hypothesis is that basically the origin of this activity is synaptic. So it has to do with something about the inputs to these cells that they're finely tuned to be balanced in a way. And we're going to test that hypothesis by including STDP in our model and playing around with the various parameters to see how that affects population statistics. So we're kind of lucky because a model like this didn't exist. So we had some fun theory to play around with, some fun computational work, but this is a very active time in this kind of modeling. And so there were a lot of pieces we could play around with and put together, although it's certainly non-trivial to get these pieces to work together correctly. So here's what we ended up with. On the left here, I'm showing you just a very, very big picture schematic of our network, right? We have about 1,000 neurons or 1,000 units. 200 of them are inhibitory, 800 of them are excitatory. The excitatory units are divided into 2 sets, one which receives inputs, which are going to be sort of our version of the cochleotopically organized stimulus inputs. And then the output units are all projecting onto one singular output unit that's going to form our decision basically for our network. This is a sort of blown up version of the network. And basically inside the network, recurrent weights have STDP acting on them as the network is training. And so there's 2 components. There's a pairwise STDP piece, which if we're looking at the excitatory to excitatory connections are basically the classic pre-post Hebbian plasticity, described by Song and Abbott and B and Poo. And then from the inhibitory to excitatory units, we have some more recent work looking at a version that I'm calling here synchronous LTP, which essentially means that synapses get strengthened as long as the pre-post firing is close enough in time, but the order doesn't matter. And if they're far in time, you get a depression of those synapses. In addition to that pairwise piece, we also have heterosynaptic scaling, which turns out to be very important on all of the synapses in the network. Now, that takes care of how the internal weights change. The external weights are shaped by what's called force training, which is a generic way of training a recurrent neural network to produce a task. And in our case, we trained our network to produce a pulse of activity when the target trial was presented and to remain at baseline when the target trial, when a non-target trial was presented. So can we get the network to perform the task? Short version is yes. Right, here's an example where it's on a go trial, on a no-go trial, and network performance over the entire stimulus set. You see that it's responding primarily to 4 kilohertz go tones, which is exactly what it should. I just want to emphasize that, you know, there's no reason that throwing STDP into a network with force should not screw up force and potentially screw up STDP to make the network pathological. So we were very excited that this worked at all. And I have some interesting results that may suggest that they're sort of more than the sum of their parts, but we can talk about that if you're curious. So first thing to check is this model consistent with in vivo observation? So first, I'm just showing you some sample traces from the network. As it turns out, this one up top is going to appear to be a responsive neuron or a responsive unit. When you look at the PSTH, the one below is a non-responsive unit. So this is how it looks when you actually break it out. Sorry, these should have been reversed. I apologize. But here's our, no, I'm sorry. This is fine. So here's our responsive unit, right? You see a burst of activity during the stimulus period and no or a lesser burst during non-target. And here you see no modulation over the trial. So it's kind of amazing. STDP gives us these. And what's remarkable is if you look over the entire network, you see percentages of nominally non-responsive cells that's comparable to what you see in vivo just by throwing in this sort of standard STDP plus heterosynaptic plasticity that people have been describing for the last 15 years. So we find that the ISIs can be decoded. So here's our original study looking at our in vivo results. The red are the non-responsive cells, the black are the responsive cells. There you can see that they're multiplex, so you can decode both stimulus and choice in all these cells across both regions. There's very few that are along the axes and only encode one variable. You see something similar in our network. So you see that the non-responsive activity actually contains information, which is what you'd hope for this model to be relevant for our work. Although interestingly, you see one difference where, although they both contain similar amounts of information for both variables, in this network, stimulus information tends to be encoded more strongly. We can talk about reasons we think that might be. So there's a lot of things you can tweak in here to look at the effect of STDP, but one of the things that really jumped out at us when we were playing around with the strength of the various mechanisms, the timescales, et cetera, although there was always effects, they were very non-homogeneous, they were very complicated. We're still trying to tease apart how they interact with each other, but one thing sort of jumped off the computer screen, and that was the role of heterosynaptic plasticity. So it turns out if you turn off or decrease the strength of the heterosynaptic terms, you basically can drop the number of non-responsive units by a fairly significant fraction. So what I'm showing you here is when you turn off heterosynaptic plasticity in the E to E synapses, you get a pretty sizable decrease. And this is across parameters for all the other terms in STDP. So this is a very generic effect, depending on, it doesn't really matter what regime you're in, it always seems to be the case. We did not observe the same for I to E. So in the interest of having 5 minutes for questions, I've shown you we can train a spiking recurrent neural network with STDP and force. We have diverse populations of neural responses that can be created using known plasticity mechanisms. Heterosynaptic scaling plays a very key role in the generation of nominally non-responsive activity. And we've got a lot to look at left. I just wanted to point out one thing on the experimental side. One of my collaborators, who's the main author on the previous study, Michelle Ancinelli, she has done some very, very exciting whole cell recordings where she's measuring both the spiking outputs of neurons recorded from auditory cortex during behavior, and then breaking in and recording their inputs. And our model is going to be able to provide some very tight constraints and vice versa on what you expect to see for responsive versus non-responsive activity, mapping their spiking activity, in other words, their classification as responsive and non-responsive, to the sorts of inputs that you can see. So that's one major thing that we hope to look at here, is if there's sort of signatures of these types of activity that we might see if we're looking in terms of the inputs. So we're very hopeful we can get some of that. So I just want to throw up my collaborators. So as I mentioned, the main author on the previous study was Michelle Ancinelli. She was formerly a postdoc at Rob Fromke's lab, and the original paper was done while she was a postdoc there. She's now started her own lab here at Pitt. So if anyone out there is a grad student looking to do a postdoc and wants to do some exciting whole cell work or some other really exciting electrophysiology or play around with the current neural network models, you should check her out. So hopefully I've got do I have like a few minutes for questions?
Yeah, we only have a few minutes for questions. If you have any questions for Badr, please ask them in the question and answer panel. This is an amazing model. It's just like impressive how it works. Yeah. Would you like to, yeah, could you please, yeah, I would like to learn it more, but I don't know if you're going to have more questions, but maybe you can start, yeah. And I can, yeah, we have a questions. Okay, I will bug you later. Okay, so Patrick is asking, what was the task the model was trained on? And does STTP improve the model's performance on the task?
So the task that the model was trained on was essentially, we basically had the same stimulus input. So there was basically a block of current that was provided during the stimulus period. It's just the subset of neurons in this input region differ depending on the stimulus presented. So you can think about it as, again, organized sort of cochleotopically. There's a different set of neurons receiving an input on different tones. And then basically it was trained so that if it happened to be one subgroup of neurons in the inputs corresponding to the target tone, you should produce a burst of activity during the response period, which immediately followed the stimulus period. So STTP, so this is interesting. So at first we were just very happy that STTP didn't break the force model because it's hard enough to get force working with spiking neurons. We didn't see a difference in performance, but what I should point out is that the performance level is fairly high with these models. We actually tweaked it so there's at least some errors. But what's really interesting is where STTP seemed to have an effect, and we're sorting out the proper controls now, is actually in robustness to noise. So if you start adding noise, either in the amplitude of the stimulus or in which neurons are actually activated. So if you occasionally randomly activate the wrong neuron, let's say when a 4 kilohertz tone is presented, the networks that were trained with STTP were much more robust to noise. They could perform better in noise, particularly in a very high noise regime. So the performance dropped, but you could still get performance out of this network even when half of the neurons you were stimulating were the wrong neurons, essentially. So, so we're investigating why that might be, but there's some good reasons to think that STTP is essentially helping to do a little bit of error correction here to keep the network active.
Yeah. Okay, thank you very much, but we don't have much more time for more questions. But if you have more questions for him, use question and answer panel to ask the questions. He will be happy to, I'm pretty sure he will be happy to answer them while we are moving to next person. And for our next speaker and last speaker for this session is Somin Christopher Song. He is doing his postdoc in the lab of Robert Franke in NYU. He will be discussing lateralized short-term and long-term plasticity of interneurons in layer 2 and 3, 2/3 of the mouse auditory cortex. I will post the link shortly in the chat, and stage is yours.
Badr, I think you need to stop sharing your screen so I can start sharing mine. Thanks. All right, so now for something pretty different. Hopefully mine's a little more wet lab based, but hopefully it'll at least whet the appetites for the modelers and theoreticians out there for thinking about what is actually going on in the brain, that at least what we can see. So my project is looking at the physiological basis for emerging evidence of lateralization in the mouse auditory cortex. And so our lab has previously shown, this is work done from a former grad student in the lab, that the left auditory cortex is important for maternal behaviors. So when the left auditory cortex is inhibited, but not the right, mouse females that are trained to retrieve pups can no longer are less likely to perform that task when it's inhibited. More recently, another former grad student in the lab, Jen Schiavo, recorded a library of pub calls and found that the prototypical pub call interval is around 150 to 200 milliseconds in between each pub call. And when played, when that library is played to naive virgins in the left excitatory neurons in layer 2-3 of the left auditory cortex show a preferential response to that prototypical pub call interval. However, once these females learn to retrieve pups, their responses are broadened to a greater range of pub call intervals. So what we wanted to find out is if there was an innate mechanism within the layer 2-3 of the auditory cortex that causes this preferential response to pub calls at this 150 to 200 millisecond pub call interval. So what I did was I recorded from chronal slices of the left auditory cortex, patched onto the pyramidal neurons of layer 2-3, and used an electrical stimulus as a crude proxy for pub calls, and stimulated them at various intervals and measured their responses. So what we can see here is both the excitatory and inhibitory currents, inhibitory on the top and excitatory on the bottom. And what we found is that both excitatory and inhibitory currents depress at all the stimulus intervals that we measured. But interestingly, the inhibitory currents depress at significantly faster rates at the prototypical pub call frequency at 150 milliseconds versus the excitatory. And this is the case in the left auditory cortex of females and is not the case in the right auditory cortex. So what we wanted to do was find out if there was a specific interneuron that was driving this specific depression at this interval. So we measured both somatostatin and parvalbumin interneurons, driving them specifically with Cre-dependent channel rhodopsin. And what we found is some disparate results. The somatostatin neurons actually demonstrated a short-term plasticity that actually increased with shorter ISI intervals, whereas the parvalbumin demonstrated depression that was similar to what was seen with the electrically driven stimulus. What is interesting is that while this may not be completely novel, Barry Connor's labs has shown similar results in layer 4 of the cortex. But what is interesting is that neither of these neurons when combined can explain the specific depression at 150 milliseconds. What is also interesting is that there is no difference in the depression that is seen in with electrical between naive females and retrieving females. So that this short-term plasticity mechanism is not probably what's causing the broadening of the responses of females of female auditory cortex after learning. So we want to look at some other long-term possible mechanisms. So our lab has also shown previously that the auditory cortex can undergo plasticity both excitatory and inhibitory plasticity through the mechanism of spike timing dependent plasticity, as shown here on the left. And then Mike Hickley's lab has shown that in the layer 2-3 of the prefrontal cortex, somatostatin interneurons specifically undergo potentiation through NMDA activated currents, whereas parvalbumin and VIP interneurons do not. So we wanted to investigate this specifically within the auditory cortex. And what we found is that yes, in my hands, somatostatin interneurons actually do undergo spike time independent plasticity, as you can see here. But when we investigated the other interneuron types, namely the parvalbumin, VIP, and then NDNF interneurons, which are used to used to label a population of layer 1 interneurons that synapse onto layer 2-3 pyramidal neurons, that neither of these populations on average show synaptic plasticity. So altogether, what we found is that the somatostatin interneurons are privileged in the mouse auditory cortex, and then they both show short-term and long-term plasticity mechanisms. And the consequences of this belies an important role in auditory perception and learning. And what we want to do is to combine these results into a modeling study to figure out what are the actual consequences in learning for the auditory cortex. So I want to acknowledge several members of the lab, including my mentor, as well as the highlighted people that are in the lab member list. These are the co-authors of the paper that has just come out about a month ago. With that, hopefully, I think I probably talked really fast, but I'm gladly to take any questions that you guys have.
Yeah, thank you, Sumin. It's just like, this is how we were planning to have more time for questions. You are the only one who did a short talk. If you have any questions for Sumin, please post them in the question and answer panel, and we will try to answer them. Did you expect, I mean, while we are waiting for other questions, did you expect these gender differences?
So I guess not necessarily the basic electrophysiological differences that we noticed. We had a hint of it possibly because of the fact that female mice showed this laterality of the responses, and then I guess we didn't expect male mice to do that as well. But we have some other evidence leading to some gender differences, but yeah, I guess not necessarily. We didn't expect to see it in the basic physiological differences.
And why do you think it, I mean, what is your speculations why this is occurring? I mean, I don't, I can't get any specific reason why why there's a big, I mean, this is a interesting differences, right?
Yeah, no, I completely agree. And I mean, it's very rare to find gender differences, as well as very rare to find laterality in the cortex, especially not in humans. So I mean, maybe it's because the female brain is, at least in mice, are need to be attuned to these pup calls, and so therefore they have to be highly sensitive to the prototypical pup call for them to be able to learn the maternal behaviors, especially if they're not being trained by another dam or another experienced female.
Okay, that's interesting. I'm just wondering, is there I have no idea about that. I'm just like wondering the number of the SST or interneuron distributions. Is there any differences between females and males?
Between females and males, I don't know if that's done actually. So that would be an interesting question. I do know that there are age differences in the expression of the different neuron types, but I don't know anything about laterality or sex differences.
This is interesting. If you have any questions, please feel free to post them in the question and answer panel, as well as our panelists. If you have any questions for Sumin, please feel free to ask them.
Yeah,
Otherwise,
I had to cut out a lot of the methodology. If you have questions about the specific methodology, I have those as backup slides if you need them.
Please go for it.
All right, so basically, for the electrical, that's pretty obvious. But for the spike time independent plasticity, the methodology I used here was I used the pre, before, post only. I have not done the reverse, but that has been shown in our lab previously to work for specifically inhibitory inhibition. But I used a 5 millisecond interval between the presynaptic stimulation and the postsynaptic current injection, and I repeated that 100 times at a 2-second interval. Waited 5 minutes and then did my follow-up recordings. And what's interesting is that, so we've found that, at least in layer 5, that the inhibitory spike time independent plasticity is also NMDA dependent. So it is not presumed that the inhibitory interneuron is providing its own NMDA. So this is sort of like a heterosynaptic or maybe non-specific plasticity mechanism.
Cool. We have one more question for you from Bernard Slater, asking for how are you doing the between cortices comparison to show that the responses are different? I see.
So especially for the short-term plasticity mechanisms, I recorded this in left, right auditory cortex, naive and retrieving females, as well as males, both left and right. And so it is only the left auditory cortex that is showing this specific differences. Males and right females don't show this laterality.
It's within the same animal, right?
Yes, correct. Within the same animal, yes. So I'll make slices, make sure to keep track of both whatever left and right auditory cortex, and then I would alternate recordings from either side to try to get within animal comparisons as well.
That sounds really good.
And thank you, Suman. If I may ask a question before we end the session as well. It's really interesting. This kind of brings a deja vu for me. Of the research that was done on the fusiform face area, as it was called, or the fusiform gyrus, as it is called now, is that a lot of us thought initially that the FFA is just accustomed to processing faces in humans, and then we went on. Researchers did more research. They found out that the FFA does other stuff in other mammals, and even other apes. It does other functions, And then they did higher, sample size research on humans, and they actually found out that, for example, furniture salespeople, they tend to process furniture in the fusiform area, and then this is to bring context for everything. And then they went on dog show hosts and dog show referees, and they actually found out that they tend to process some things like canine teeth or some very specific things that they were highly specialized in that area. And right now, there's more body of research that actually shows, fusiform area is for specialty, and us humans, as social animals, we tend to focus a lot on face processing. So in smaller communities, you might not have as much focus on FFA for that. So would this difference, this lateralization, would that possibly be something like this, that it is actually the result of a flexible environment in the cortex that actually has given rise to two different behaviors physiologically? Would that be possible in your opinion?
I mean, yeah, I would say that's possible. I mean, we do know that for mice, vision is probably the least of their sensitive senses, and therefore least used in the processing. So, but hearing is a lot more sensitive, and therefore would probably belie more specialization in their processing, and so therefore maybe you have more lateralization in that case. Yeah, I mean, it is definitely possible that especially for females that, like, in order for their species to survive, need to be able to care for their pups, and therefore they need to be able to respond specifically for these pup calls. And females that are not trained to raise pups need to be, vigilant for these prototypical pup calls.
I think at this point we should just summarize, and thanks to Sumin, Christopher, it's just very nice work and it's interesting findings. And I also would like to thank to all of our speakers, Melanie, Badr. It was really beautiful results, and I can see your reason why you made the really long talks, because you wanted to share your interesting findings. And I also would like to thank to our audience for joining us in this session. I think we should also thank to Telha for giving us technical help and sharing our session during the session at the Twitter and making it much interactive. I hope you enjoy these sessions and hope to see you in the next NeuroMatch conference. And thank you very much, everybody.
Thank you, everyone. I'm gonna end the talk in 5 seconds.
Thank you.
Thank you.
Bye.
Astan Shamorat, I hope I pronounced it correctly. Her talk is titled Paired Stimulation in the Human Limbic System, Induced Functional Connectivity Changes Consistent with Spark Timing Dependent Plasticity. So the stage is yours, Jasmine.
So hi everyone, today I'm going to talk about our work on pair stimulation in the human limbic system. This work is done at the University of Washington and in collaboration with Dr. Edward Chang's lab at UCSF. As you may know, many neurological disorders can be attributed to abnormal neurodynamics at the network level, which comes from problematic neural connectivity. And this can range from stroke and traumatic brain injury to movement disorders like essential tremors, as well as some psychiatric diseases. Meanwhile, recent work showed the exciting potential of engineered plasticity, which involves using spatially and temporally specific neuromodulations to reorganize selected connections. With more ongoing research, this kind of targeted reorganization may serve as a potential therapy for these different brain disorders. One hypothesis of achieving this kind of engineered plasticity is using pattern stimulation. Numerous studies have demonstrated that electrical stimulation can either strengthen or weaken neuronal connections. A recently proposed mechanism of this is the STDP rule, which suggests that connections between two neurons can be strengthened when a postsynaptic neuron fires shortly after the presynaptic neuron within up to 20 milliseconds. And when this happens repeatedly, showing this red curve here, It leads to increased synaptic weights or stronger connection between the two neurons. So there are several stimulation methods that people have used in the past to change neuronal connectivity based on STDP, including activity-dependent stimulation and pair pulse stimulation. So in our study, we analyzed the network effect of pair pulse stimulation in the human brain. Because although strong evidence of STDP has been found for synapses in vitro, there are still lots of uncertainties around its effect in vivo, especially in the human emotion processing regions. Therefore, this is the first study to test the concept of STDP using clinical data from these regions and to understand the changes in functional connectivity as a result of paired pulse stimulation. Our data used in this study are collected at UCSF in our collaborator Dr. Edward Chang's lab with the help from Kristen and Ankit. Patients have different numbers of ECOG grids or depth electrode implanted in the limbic system and related regions shown here. A few examples include the hippocampus, amygdala, and orbital frontal cortex, which are known to be involved in emotion processing and decision-making. We paid particular attention to the orbital frontal cortex here when designing the experiment because previous work from the Chang lab suggests that electrical stimulation of the lateral OFC improved mood in patients with depression. Therefore, we paired OFC with other emotion processing regions during stimulation to see if we can induce targeted changes between them. Here, 10 milliseconds and 100 milliseconds time delays are used between selected regions for each recording block. If the stimulation-induced response does follow the STDP rule, we'll see a distinct difference between how these two paradigms change the functional connectivity. So to measure functional connectivity between brain regions at the network level, we computed the coherence between each pair of electrodes. Coherence has become a very popular metric of functional connectivity during spontaneous activity because it measures the phase synchrony between two signals at a given frequency. For example, if we take these two LFP signals collected at channel A and B, the connectivity between their respective recording sites can be estimated from the computed coherence. Here's a brief summary of our experimental design. So we used 10-second segments of the baseline recording between simulation blocks to compute coherence and estimate the changes in functional connectivity. So based on the principles of STDP, our hypothesis was that baseline coherence will change more after 10 trials of stimulation with 10 milliseconds delay compared to 100 milliseconds delay in the control. So I'm going to talk about two types of coherence quantification today. The first one is shown here between each pair of electrodes within the stimulated regions. And the second one I'm going to cover later is between all recorded regions to look at the whole network. You may notice here that when we are selecting two regions for stimulation in each patient, OFC is actually common for all of them. And this choice is based on the work from Chang's lab, as I mentioned before, which showed that stimulating the OFC leads to mood improvements. And with this experiment and some future work, we hope to correlate the functional connectivity measures with mood assessment to further understand how paired stimulation affects the emotion processing circuits of the human brain. So to go into some preliminary results, we first look at three traditional frequency bands, delta, gamma, and high gamma. So for the lowest frequency band here, the bar graph on the left shows the coherence value, and the first baseline and the last trial after stimulation for short, long, and control sessions. So traces on the right shows the changes in coherence over time in between simulation blocks. We saw that for delta coherence, short delay induced more consistent positive changes, while control and long delay oscillates around 0. For gamma coherence, only short delay induced significant positive changes in the bar graph, and a similar trend can be seen from the overtime plot. For high gamma coherence, short delay again induced the largest positive changes, while control and long delay sessions oscillates around 0 over time. So apart from these traditional frequency bands, we're also curious about what's happening at other frequencies outside of these defined bands. So we look at coherence across frequencies more systematically. We summarize the changes across all possible frequency bands into the heat maps shown here. So only changes that are significantly different from 0 are colored in these heat maps. So these preliminary results suggest that 10 milliseconds delay induced the most positive increase in coherence comparing to 100 milliseconds in control, especially in the 50 Hertz to 80 Hertz range. So in the second part of this analysis, we expanded our approach and quantified the coherence between each pair of electrodes within all regions using the same method. Results from all 4 patients here are pulled together. Again, for delta coherence, short delay induced more consistent positive changes, while control and long delay sessions oscillates around 0. For gamma coherence, short delay induced stronger positive changes, but long delay sessions also induce some significant increases in coherence over time, while control sessions stay very minimal, close to 0. For the high gamma band, both short and long delay sessions have led to an increase in coherence, and they oscillate across time. So one possible explanation for this observation is that gamma and high gamma oscillations in the brain represent more local information. And that's why when we're expanding to look at the entire brain and all recording sites, we don't see a clear trend in them. Again, when we expanded our analysis to all frequency bands, we see more clearly that in most frequencies that's lower than 80 Hertz, short delay stimulation induced much stronger increase in coherence between all brain regions compared to long delay and control. So these preliminary results show that both stimulation paradigms significantly alter the functional connectivity between brain regions compared to control. Meanwhile, pair stimulation with 10 milliseconds delay led to greater increase in LFP coherence compared to that with 100 milliseconds delay across multiple frequency bands. So these results together support the role of an STDP-like process in the emotion processing regions of the human brain. For future work, we plan to collect more data from additional patients with very similar electrode coverage, counting the OFC. And we're going to analyze cross-frequency coupling, which represents interregional communication in the brain. And hopefully, we will carry out mood assessment during the pair stimulation paradigms to understand their emotion correlates in the brain. I want to thank Kristen and Ankit at the Chang Lab on collecting and organizing the data, and our previous rotation students, Kira and Sam, here for some pre-processing. And of course, my advisor, Dr. Azza Yazda, and all lab members at the Yazda Lab. Thank you, and I'm open to questions.
Thank you, Jasmine. It was a very nice talk. Thanks a lot. Let me check whether we have questions. So you can post your questions to the Q&A panel.
Can I ask a question?
Of course, sure.
I haven't worked with like human, but I know that in STDP type of experiments in a slice, it's important what is the baseline level of your stimulation, and when you want to pair it, if it's like below a threshold, it's not going to have a big effect. How do you come with the intensity of your stimulation besides the timing?
Yeah, it's a very good question. So for, let me go back to the, here we go. So for our current, it actually depends on the patient's tolerance for clinical data. Because these patients have the implanted electrodes for seizure monitoring, not particular for this experiment. So we are collecting data on top of seizure monitoring. So we can only apply the current that they can tolerate for each patient. Yeah, so that's like a constraint of clinical data collection.
Thank you, Ferizan, for the question. Any other questions? Maybe I can ask one then. That might be a naive question. So you were talking about short delay and long delay stimulation being different from each other. I mean, maybe because I'm not so familiar with the topic, but what would be the impact of short and long delay stimulation, or what does it tell us?
Yeah, so we picked the 10 milliseconds and 100 milliseconds as the short versus long to test STDP because more previous work on slides and in vitro data suggests like this nice trend. When you are below the 20 milliseconds, you see long-term potentiation, and when you are above 50, you see very minimal changes. That's why we kind of picked 10 and 100 to separate them to be far apart. So if they follow STDP, we see changes for the 10, but not for the 100.
Okay, thank you. Thanks.
Sorry, I also have a question.
Sure.
Are you going for mechanistic approaches, or have you also considered Bayesian non-parametric search for the right parameters? Because it's quite hard to try different delays and different parameters.
Yeah, that's a really good question. We haven't actually, from earlier NeuroMatch talk today, it kind of inspired me how they kind of pick the parameter, because it is really hard to choose the right frequency and like to induce the most changes. So maybe we can optimize this process more to know what's the optimal simulation frequency. Now it's at like the burst frequency, and other parameters can definitely impact the results.
Thank you.
Thank you also, Said, for the question. So if there are no more questions, I think we can go on with our second speaker. Okay, our second speaker is Mahima Sharma. She's a postdoctoral researcher at the Department of Biomedical Engineering, City College of New York. And she's working with Lucas Parra. She's also an advocate of mental health and equity in education. And her talk today is titled Synaptic Evidence for the Cumulative Effects of TDCS in Spaced Learning.
Thank you, Ezgi, for the nice introduction. Hi, everyone. I am Mahima. I'm working in Professor Lucas Parra's lab, as Ezgi mentioned, in the City College of New York. And today I am going to present exciting results from our lab on the synaptic evidence for the effects of TDCS in the space learning in the hippocampus. So let's move on with the topic today. So transcranial, transcranial direct current stimulation, also popularly known as TDCS, is a non-invasive brain stimulation technique where a weak constant current of 2 milliamps or less is applied across the scalp. So the current enters from the anode and it exits the scalp through the cathode. And this application of current generates an electric field between the two electrodes. And this electric field is usually in the range of 1 volts per meter. And what does this electric field does? What it does is that it leads to polarization in the brain. So certain areas are hyperpolarized and certain areas are depolarized. And Farzan, who is going to talk right after me, is going to talk more about the polarity and TDCS. So this simple technique is very effective in different forms of improved learning in humans, and it has been shown to be effective in various conditions like neurorehabilitation, psychiatric disorders, and cognitive enhancement. So stimulation is usually applied during a training regime, and then a lasting enhancement is seen, which is specific to that learned task. Now, it's great that TDCS works better in human trials. We see its effectiveness, but we are still do not really understand what are the underlying mechanisms of TDCS, which can help inform the future protocols for TDCS better that can concern the safety and the efficacy of the TDCS. So how do we do that? So animal models allow us to study the underlying mechanisms, how exactly the TDCS is causing the changes in the neuronal network and what are the consequences. And for that, of course, like there are many in vitro studies which point that TDCS improved the learning by mediating the Hebbian synaptic plasticity. So these are the recent results from our lab where Greg Kronberg led the study, and what he found, as you can see in the red here, red is the anodal stimulation of direct current stimulation to the hippocampal slice, which was when paired with the TBS induction. It resulted in a boost in the synaptic plasticity as it evident in the red when you compare the black and the red. So now there are many in vitro studies which support this theory that TDCS improves learning by modulating Hebbian synaptic plasticity. But there are two main challenges with that study. So what are those challenges? The first challenge being that the electric fields which are used in these in vitro experiments are much higher, in the range of 20 volts per meter, as you can see here, than the electric fields generated with the TDCS in humans, which are in the range of 1 volt per meter. And another reason why the effectiveness of TDCS has been called into question is because of the relatively small effect sizes, as you can see here. Like you see there's an effect, but the effect size is so small that we have to do many experiments to really reach the significant level. So we know that in humans, we do see an effect of TDCS. So definitely something is happening in the synaptic plasticity level. But of course, we are missing something in the protocol, or some point is missing, which is preventing us from really dissecting out the mechanisms. So that is what we are trying to figure out. And that is what the objective of our study is. How do we resolve these two challenges so that we can study the TDCS better? We can really dissect out the mechanism, which can inform the future stimulation protocols, keeping in mind the safety and the efficacy of TDCS. And also in the same study, the one part of the study was a dose response curve, which we did, which showed us that the higher field intensities will have stronger effects, as you can see here. But again, the effect size is very small. So our goal is to how do we resolve these effect sizes? So in order to get there, what we did is we did some research and we looked back into the human TDCS domain. And what we are seeing is that there's much more popularity of this multi-session TDCS. So where did this multi-session TDCS came from? So the scientists who are working with the human TDCS domain, they have leveraged this observation that effects of learning in humans are known to accumulate over multiple bouts of learning, which is known as space learning. And then deriving inspiration from the same, they are doing multiple sessions of TDCS paired with training as spread out over different days and multiple sessions. And what they have found is that the effects are much more stronger, whether it be in case of motor rehabilitation, language learning, or other cognitive enhancements. So now we have also seen that there is a synaptic evidence of the space learning in the hippocampal slice. So there are in vitro studies. So here, this is an example of a study from Gary Lynch's group, where they have used TBS, theta burst stimulation, to induce LTP and see how the space learning effect is there. And as you can see, there's a cumulative effect. So now, having known that, it led us to hypothesize that maybe, to resolve these challenges which we are facing in the in vitro study for the TDCS and to improve the effectiveness of TDCS at realistic field intensities, maybe our answer lies in the space learning paradigm. Because in space learning paradigm, we see the cumulative effect. So maybe TDCS will have a cumulative effect in the space learning paradigm, which will help us resolve the effect sizes, one. And second, we can also maybe go with lower field intensities, which will be realistic as compared to the human studies. So these are our two hypotheses. The first is that the effect of DCS on synaptic LTP could accumulate over time in a space learning paradigm. And the second being that the stronger fields should lead to bigger effects. So now how do we check these hypotheses? For that, we have taken male vista rats, which is 25 to 30 days old. We made 400 micrometer thick hippocampal transverse slices, which were incubated in interface chamber for 30 minutes. Before that, there was a recovery in the holding chamber, 30 minutes each. And then we place these hippocampal slices in a parallel orientation between the two AGCL wires, which are parallel to each other. These are the wires which are used to apply the direct current stimulation to these hippocampal slices. And then this is the location of the electrodes. We have a stimulating electrode for orthodromic stimulation here. And then there are two electrodes. One is in the dendrite, another is in the soma. So then dendritic electrodes picks up the field EPSP signals, and the soma electrode picks up the pop spike signals. And then of course, after that, this followed by taking input output curve, a baseline recording, and then TBS stimulation in coupled with the DCS. So what did we find? So here are the results. So we have used the TBS protocol, which is 4 pulses at 100 Hertz, repeated for 15 bursts for 5 Hertz. So this is a high intensity TBS, which is being used in our lab in the past in vitro studies for the TDCS. And what we have found is that when we couple the TBS space learning paradigm with electrical stimulation of 2.5 volt per meter, again, we do see an effect. There is an effect. And then we thought, okay, this is 2.5 volt per meter. Let's go to 20 volts per meter and what happens? And this is what happened. So we do see an effect, but the 20 and 2.5 really overlap. And also between the control and the anodal, there is not a significant difference, as you can see here. There's a trend, there is an effect, but there's not significant difference. So again, we came to square one. We couldn't resolve the effect size. And again, the induction was too high. We couldn't see a stronger effect at high field intensities. So we had to really pick our brains and figure out what is the problem. And then we realized the problem was in our intensity of TBS, because we are using the maximum intensity TBS. So it's equivalent to me performing my best. And if you apply TDCS to me, I won't be able to perform any more better because I'm already at my best. So then we look back at the review and we realize the space learning studies are done with the 10 bursts of TBS. So we decided, all right, let's go to the 10 bursts of TBS, and maybe then we will be able to resolve our questions, which we have been facing for so long, and we could improve the effectiveness of TDCS. So we moved on to using 10 bursts of TBCS, 4 pulses, and then again, the TBS, we applied 4 inductions, and when we coupled these TBS, this time we used electric field of 10 volts per meter, and you can see that we could really resolve the effects, and at each TBS induction between the control and anodal, there was a significant difference, as is evident from here. So here, not only we were able to resolve the effect size, but we also saw that the effects are much more larger and they accumulate over time. So these are the conclusions from our study. So the conclusion being that DCS applied during repeated bouts of TBS resulted in an increase of LTP, and the space learning was saturated quickly with the stronger TBS protocols. Therefore, it was obscuring the cumulative effects of DCS. But when we used a weaker TBS, we could see cumulative effect of space learning, including a cumulative boost of LTP with electric fields of lower intensity. So the take-home message for all of you guys is that, sorry. The effects of TDCS accumulate through an increasing synaptic strength after repeated bouts of learning. So that's all from my side. I would like to thank my PI Lucas Parra, the lab members, the Neural Engineering Lab Group at CCNY, NeuroMatch, for giving me the opportunity to share my results, and of course, the lovely audience we have here today, and I'm open to questions.
Thank you very much, Mahima, for this wonderful talk. It was really nice. So do we have questions? You can again post the questions at the Q&A section. Okay, so maybe I can ask my question then. What would be the because I mean I'm always thinking about like human research. So what would be the implications for using TDCS in humans?
So the TDCS is already being used in humans. There are many investigations which are being done for its effectiveness, and they have found it's effective. It's just that the challenge we are facing right now that we want maximum effectivity at lower intensities, because we have to take safety into consideration. So there are certain human studies that use different types of electrode montages to generate an electric field of 6 volts per meter. So this studies aim to like how we can, while applying lower field intensities, still have a maximum effect of TDCS so that it's within a safety threshold, and whatever questions which are being asked and for the effectiveness of TDCS, we could address that question. So that's the purpose of this study.
Thank you. Thanks. Okay.
I'll ask a question.
Yeah, sure.
Do you see this will be very easily translatable to human study, because they can just apply space learning and with the same parameter, but just adding in space learning to like a clinical study?
Yes, actually, like the inspiration also came from the human studies, because they are doing the multi-session TDCS session, and every time they are doing a training coupled with the TDCS, because again, there are different ways they apply TDCS. Is it like online or offline? Is it during training or before training or after training, and then they see the effect? So in our study, of course, we are interested to know the online, like what happens if we are applying the TDCS during the LTP induction, and which can be translated into humans when TDCS is being applied during the training sessions. And it is showing that it is effective, but what our aim is to like really figure out what are the underlying mechanisms, so that we can fine tune the protocols which we already have in the human domain, and also like really work on the safety and the efficacy, because those are the two important components which are really necessary for any intervention which we need to work for the humans.
Thank you. Sounds good.
Thank you also, Jasmine, for the question. So maybe one last question. What would be the next step for you?
Oh, so the next step, yeah, I forgot to mention in the excitement. So next, as I showed that we have done the study for the 10 volts per meter. So my next step is to check with 2.5 volts per meter, because again, we want it to be at more realistic field intensities, because we already see a very significant effect with 10, which is already half of what we were using before 20. So now our next step is to do it 2.5, which we did with the higher intensity and we saw something. But now, since we know that with the lower intensity TBS, we're able to resolve the effects using the space paradigm, that is our next step. And of course, in the interest of time, I wasn't able to share other observations from the same data. Like we do see a difference between the induction and the maintenance, and the TDCS is affecting the maintenance. So of course, asking that question would be the next step also.
Thank you. Thanks, Mahima. Are there any other questions? I don't see any. So then we can move to our next speaker. So the final speaker today in the session is Foruzan Farahani. She's a PhD candidate in biomedical engineering at the College of New York, and she's working with Lucas Parra. Her talk is titled Modulation of Synaptic Plasticity in Single Neurons with Transcranial Direct Current Stimulation. So the stage is yours.
Hello, everyone. Today I'm going to talk about modulation of synaptic plasticity in single neurons with transcranial direct current stimulation. So Mahima talked about more in detail about what is transcranial direct current stimulation, but just shortly, it's an non-invasive brain stimulation method that they put 2 electrodes over a scalp and they pass through a certain amount of current. This current will generate an electric field across the brain. And what is interesting for us is to know that how this electric field is affecting a single neuron. Based on computational studies, we know that there's going to be a gradient of polarization across a neuron, meaning that one side of a neuron is going to be depolarized while the other side is more hyperpolarized. Knowing that what's happening in the level of a single neuron computationally, let's go back and then look for the debates that what is the most effective way of using TDCS? Is it better to pair TDCS with the training, or it's better to apply TDCS before or after a training? To answer this question, we have been using hippocampal slices and using theta burst stimulation to see that whether it's better to pair the electric field with the induction paradigm or not. A study published by a previous member from our lab, Greg Kronberg, has shown that pairing DCS with theta burst is able to modulate the synaptic plasticity, and anodal DCS is capable of boosting this synaptic plasticity. So the question maybe is like, what is the underlying mechanism of what Greg has been reported? So we know that based on Hebbian plasticity, neurons that fire together wire together. So having this rule in mind, we may think that if we apply electric field during the induction protocol, we may push the postsynaptic neuron to fire more. That means that we are going to have a bigger synaptic plasticity. So to test this hypothesis, I've been using preparation that is more suitable for whole cell patch clamp recording, usually in studies that they are trying to investigate LTP in patch clamps. They usually apply GABA blockers, and that needs to cut the CA3 area of the slice in order to prevent seizures like activity in the brain. The rest of the setup is very similar to what Mahima talked about in the previous presentation. So I will go and talk about what was the result of the experiment. So this is the protocol that I used to induce the synaptic plasticity. It's slightly different than the one that Mahima described. I tried to use this one because it's the common protocol that people use in wholesale patch clamp recording. And as you can see here, this is a trace of neural activity during the induction. And you can see that this induction is capable of making the postsynaptic neurons to fire. So what was the results? The results showed that anodal DCS is capable of inducing a higher amount of potentiation. And at the same time, we saw that there is a correlation between the number of spikes that a neuron exhibit and the amount of potentiation. So by now, everything is like aligned with what we had in our hypothesis, that we are going to apply electric field, we are going to have more spiking in the postsynaptic neuron, and then we are going to have higher plasticity. But then we wanted to dig deeper and see that how much this postsynaptic spiking is important in inducing more LTP. The first experiment that we had in mind was using a voltage clamp recording. In that way, we will clamp the postsynaptic neuron to have a certain potential, and we can test that if this is what we expect to do. Unfortunately, we soon realized that voltage clamp recording is not a suitable way of experimenting, as DCS introduces an artifact, and we were not able to remove that artifact. Then we decided that, okay, let's make this neuron not to fire. And then we used a sort of a blocker inside the intracellular solution, which is called QNX. But then I realized that in order to block all the spikes, I have to use a very high concentration of QNX. So that made the recording to be really unstable. So what we end up to do at the end was using current injection and trying to emulate the effect of DCS in the soma by injecting the current. So that we considered two different experimental setups. In one, we injected a depolarizing current and made the soma of the neuron to be about 3 millivolts more depolarized than the resting state. And the other condition was, while having anodal DCS, we will inject the hyperpolarizing current. In this way, we sort of cancel out the effect of DCS on the soma. Then what we saw as a result was, indeed, again, we had the more spiking and the more potentiation. and like results seem to be aligned with what we expected, but we saw something unexpected when we compared depolarizing current injection and anodal condition. While depolarizing current injection had higher potentiation amount, we saw that anodal condition has higher number of spikes, which means that in an anodal condition, we had more spikes, but we had less LTP compared to depolarizing current injection. So this was sort of puzzling for us why this is happening. Then, fortunately, I had a lot of recordings that I ended up not using them in my paper, but they were helpful to solve this sort of problem. I saw that in some of the neurons, when I block the GABA, the moment that the electric field goes on, I see a spontaneous activity before starting the theta burst stimulation. While this behavior was absent when the GABAergic input were intact, or when the intensity of electric field was lower. So we thought that maybe DCS is inducing sort of a spontaneous activity in the neurons, and this may sort of affect what we are seeing in the LTP. So in order to sort of bridge what we are seeing here in as a spontaneous activity to LTP, we started to use a computational model. Again, Greg Kronberg in his publication has shown a detailed morphological model in order to combine the effect of DCS and synaptic plasticity. What I've done was adding a parameter for scaling synaptic plasticity. This was due to a previous publication showing that a network burst activity can modulate the amount of synaptic plasticity, and this is due to the changes in resources available during the synaptic plasticity. So I used what I've explained shortly here in my computational model. This here you can see a sort of a visualization of how membrane polarization looked like in my model in these three different conditions. And then I tried to do the modeling for showing the effect of this spiking, especially the spontaneous network activity on LTP. So I was able, with the model that I suggested, I was able to capture the effect both on the number of spike counts that I had, and also I was able to show the effect on the LTP. So this shows that this spontaneous activity is sort of acting as a homeostatic mechanism and sort of limiting the amount of potentiation that we expect from anodal DCS. So at the end, what we saw in this study was, first of all, we had like a direct evidence of how DCS is affecting neural excitability and how that can change the Hebbian synaptic plasticity. At the same time, we realized that DCS can induce a spontaneous activity when GABAergic inputs are blocked. And this spontaneous activity can sort of trigger a homeostatic plasticity mechanism. And at the end, our result is a mix of Hebbian plasticity and a homeostatic plasticity. At the end, I want to thank my PI, Lucas Para, people in our lab, especially Greg Kronberg, that was very helpful during this study, and other people in a neural engineering lab. And I'm ready for if you have a question.
Thank you, Feruzan. It was a very nice talk. Thanks a lot. So you can post your questions on the Q&A section if you like, or on the talk, the chat, sorry. Okay, so for now, I don't see any questions, so I can maybe ask my question. So you mentioned that you observed a spontaneous spiking activity right before stimulation. That was, I mean, you also said you wouldn't expect something like that, but maybe can you elaborate a bit on that?
Yeah, previous study, one study that we had in our lab, they were doing experiment in cortical slices, and another study in another lab that they use hippocampal slices. They showed that applying an electric field over 30 volts per meter may induce a spontaneous activity, while I was using an intensity that was in lower bound of that threshold. So I didn't expect that with 20 volts per meter, I will see a spontaneous activity, especially having in mind that based on our model, 20 volts per meter in a slice, which is like silence, it's inactive, it's not like a human brain, which is like in active state. It's very far away from the threshold for spiking. And 20 volt per meter is about, it generates about 3 millivolts maybe polarization in a pyramidal neuron. So I was not expected with 3 millivolts of polarization, I get to make like a burst of activity in a neuron. So that was like sort of surprising and wasn't expected for us.
Thank you. We have one other question from Max Korbmacher. So Max says, thanks for the interesting talk. To which degree do you think do you think your findings generalize beyond the hippocampus?
That's a very good question. One thing that makes hippocampus and this Schaefer collateral and CA1 sort of secretory very suitable for our studies is that the way that presynaptic neuron and postsynaptic neuron are organized. So in this way, we are sure that like we probably are not affecting the presynaptic input directly. But I think that knowing that what it's going to happen when we have a more controlled system, we can better generalize it in other area of the brain, like cortex, which we are interested to do experiment in, but it's more difficult to interpret the result when electric field is not only affecting the postsynaptic neuron and it's affecting presynaptic neuron at the same time.
Thank you. Also, thanks very much for the question. And we came to the end of our session. I would like to thank all of you again, the speakers for the wonderful talks, and also the attendees. And special thanks to our backend, Saeed Salehi, who's been doing all the technical work. And yeah, enjoy the rest of NMC and have a nice day or evening. Bye, everyone.
Thank you. Bye.
Bye-bye.
Thank you. Bye-bye.
Yeah, thanks.
Bye.
Thank you.
He drew all of these pictures by his hand, like staining neurons that he did in his lab and look at under microscope. And now, a century after that, we have a bunch of tools and techniques to image from very coarse resolution to nanometer resolution. And we even are able to picture the whole brain connectome. And that has opened the door for us to study the structure, the geometrical structure of the brain. Okay, so what is the neuromorphologies? And if you haven't heard about neuromorphologies, I have actually plotted a few of them, 6 different cell types here that are, for example, from omicron cells that are in the retina cell, in the retinal area of the brain. These are all from the mouse brain, or perconjugranal cell from cerebellum that are working on the motor system, or different pyramidal cell in different layers of neocortex. Even we have a good converged way of like putting all data together, and one of the good successful one is neuromorpho that has more than 100,000 neurons of 50 different species. So we are able to look at the variety of the cells in different brain regions and species. In my opinion, there are two main questions when we see neuromorphologies. The first one is that how we can describe this geometrical structure. What is their geometrical feature? What generative models make them? And this is kind of a question that relates to the cell type identification. If we want to find, for example, new cell types, we have to say, based on the morphology, of course, we have to say that what kind of geometrical feature we expect, or developmental studies, that is the neuron is changing over time, or even larger scale simulation of the brain that we want to make a big like simulated version of the brain. And the second question is that given the morphologies, what kind of function we expect? In other words, what kind of what kind of function we can see when we have this geometrical structure? This is a question that relates to the physiology of neurons. We know that a simple sigmoid function of neuron is not enough to describe the function, but then what is the best way to describe them? Or the other thing that is related is the biological plausible neural networks, how we can incorporate these morphologies in the neural networks. Okay, so let's go from the morphology to a little bit simpler structure. Here I have plotted one neuron that's connected possibly to a few other neurons in the synapse with different synapse locations. And in the dendrogram structure of this morphology, I ignore all the geometrical structure and just keep the branching order. For example, I would start from soma and see that where is branching and add a branching to this diagram here. Even I can see that where are these connections or where are the synapses on top of that. Okay, so it's a simpler version of the morphology. We are ignoring the all 3D structure of that. But still, it's not very simple to study. Here I have another like Purkinje cell and its dendrogram that is sorted different based on the depths of the distant dendrogram. We can use, for example, a simple motif analysis to see that within this graph, what kind of branching we see. For example, here I can have 3 different branchings. The branchings that both of the children that it has are leaf, like for example, all the branchings around the end of these 3, or the branchings that one of them is leaf, the other one is branching, and the branching that both of them, like the red one, both of them are branching. And you can count the number of them with different depth and see that how this profile changed from one cell to another cell type. This is, for example, for the Purkinje cell. You can do for the bigger motifs, these are the motifs I would call them, and you can sort them and kind of differentiate different cell types. For example, for a few cell types, for apical dendrites, basal dendrite, granule cells, ganglion cells, we can count that how many motifs of order 8, by order 8, I mean 8 roots, 8 leaves at the ends, how many of them we see in the single neuron? Or if we average it on average, how many of them do you see that? And you can see this trend to differentiate different cell types and see that are these 2 classes different or not. But there is a big challenge when you do this analysis. First of all, it's a simple analysis, it doesn't give you good intuition, but the main challenge is that this number of motifs, if you a little bit go further than 3-4 nodes, they grow super exponentially. For example, if you have 4 nodes here, you just have 2 different topologically different trees. If you have 5, you have 3 different ones. But if as soon as you go to the higher and higher number of leaves, you have more and more number of motifs that you can generate, and it doesn't give you a good way to sort them. Okay, so here in this talk, I want to give you a simple model that describes the morphologies. Let us start with the simplest branching process that we can think of. In the simple branching process, you have a probability distribution, let's say P, and based on the P, each node it may branch or terminates. And this probability P can depend on the depths that you go. This is, for example, one typical example. If you have the p is equal to 0.045, and even you can, given this p, you can look at the likelihood. This is a generative model, so we can compute the likelihood, and the likelihood only depends on the branching at leaves. That's a very simple model, simplest models that we can think of. But we know that's lacking a bunch of information. For example, if you look at the neurons, in most of the cases, you see that there is a main branch that's coming out of soma or somewhere, and then there are some small side branches coming out in different locations. If you see under developments, you see that this main branch that are usually thicker, they stay, and they just like have small things coming out, retract, and they continue growing. Okay, I want to add this feature to the simple model that we have. It's actually very easy. I'm going to extend the branching process that I showed you. Let's assume that we have two different types, let's say A and B. A correspond to the main branch and B correspond to side branch. And you have two different probability distribution that possibly these PA and PB are different, that if you are in the A location, A node, you have the probability PA to branch, and if you are in the B nodes, you have the probability of PB to branch. If PA is very high, for example, in the case that you are in the main branch that's wants to continue, and PB is very low, you have a typical structure like this one, that the main branch is trying to survive and the side one is dying as soon as it comes up. Okay, great. So after we wrote this model, it's easy to generate the samples here. But one hard problem that we had was that given this model, how we can calculate the likelihood of each tree? Like given this tree, for example, and P and PB, what is the likelihood of seeing that? After a bunch of trial and error, we found the formula in this case. And the formula that is here, if you look close at this formula, you see that there are a few more parameters, n plus minus, n plus plus, and n minus minus, that if I flash back to other slide that I showed you this one, this is actually the number of these values in different locations. Okay, great. So we have now a model, we have a generative model, and we have a likelihood of them. Then next is to try in different cell types, see that what we observe from different cell types by applying these models. Okay, so fitting on the cell types, we observe that that is actually a very typical structure that when we try different cell types, we observe that for example, let's say in apical dendrites, you have a point that P and PB, these are a graph, the blue one is the PA, the orange one is PB, and this green one is the simplest model that I presented at first. And I'm fitting on different depths. Okay, so when I apply on, for example, this subclass, the apical dendrites, for example, I see that at some point they fork and PA become higher and higher, PB is becoming lower line. It means that in this area, the neuron is very asymmetric compared to this area that's probably very symmetric growth. And this pattern repeats almost on all the data set that we had. That was interesting for us. That is a general phenomenon. Okay, great. So we have a thing, we have a model, we have fitted on the cell types to see some profile of the parameters in these models, but how we know that these models are superior? We have likelihood, and this likelihood tells us to compare different models. For example, for this case, we can simply compare them with the symmetric one that has one parameter, or the asymmetric one, P and PB that I told you, the one that are depth dependent, or the one that are not depth dependent. And the general message that we learned was that that's actually true, that the asymmetric one is actually superior when you fit on the dendrogram. Great, so this is the end slide. I'm going to just give you takeaways. We have three different takeaways here. One is that we can use motifs to compare different cell types. And this is a way to distinguish different cell types or possibly find the new cell types. We present depth-dependent asymmetric models, and we observe that superior to other models. And when we feed, we realize that at the beginning, they are very symmetric structure, and in the longer scale, they have very asymmetric. And thank you so much for joining us. I want to acknowledge a few people that helped me for these projects. Konrad Kording, that he's a PI, and as you see, he's very much into the CPUs and neuromorphologies. David Rolnick, he's right now in the Professor and Mila, and that is basically the work with him. Khashayar, we have worked on the functional part that I haven't talked about that. If you have time, we can go through that. Lena, that she's actually presenting a talk very soon after here, and Devang and the rest of the coordinates. Thank you so much, and I am here for any question that you have.
Great.
I don't know how much time we have.
We have 1 minute-ish left. Thanks, Roosevelt, for the great talk on this interesting analysis with the dendrogram of the neurons. One Q&A question we have is, how do you encode the depth? How do you encode the depth dependence on the model? To further clarify, is there some sort of functional form for how PA and PP change with depth?
That is very good question. Thank you so much. Okay, so I had actually 2 bonus. I can go quickly to these 2 bonus. One of the aims that I have is that to simplify the structure of neurons. For example, if you have this one, you can say, okay, that's a different compartment that it has. And this comes back to the structure that I told you. If you have this simple structure, one observation that we had, and that was a thing that still we are studying, that is that you can try different structure, different, for example, trees that, like, for example, different trees with different networks that are associated with each nodes, and see that how well they perform on different tasks, like let's say CFAR task. And the thing that we observe, and that's kind of our counterintuitive, is that, for example, for CFAR task or for image classification task, when you have more symmetric one, you have a better chance that you train on the data set than you have asymmetric one. It's a kind of reverse, like fix that. But that's a question that if you have different structure, what kind of data set it fits better? That is actually a question that we are still studying on that. There is actually a next talk after here, Elena. She's talking about the similar stuff if we are interested in that.
Great, thanks for the question. Thanks for the answer. I hope that solves the question from Daniel in the audience. Okay, yeah, given the time, we probably need to move on to the second talk. And thanks to Roosevelt again for the great talk. Next, we have John-Woo Tron, who is a grad student from Joshua Wagenstein's lab in Johns Hopkins University. Today, he's going to talk about human structural connectomes are heritable. Okay, John-Woo, why don't you take over the lead and unmute yourself?
All right, can you hear me? Okay, great. So today I'll be talking about structural connectomes and their heritability of them. So what is heritability? Let's see the variation in phenotype or phenotypes due to variations in genetics. And it's interesting because one can potentially discover relationships between diseases and genetics. And in neuroscience, one thing that we might be interested in is understanding how the connectivity of the brain itself is influenced by genetics. So we're going to ask the simplest possible question, and it's that, are the connectivity patterns heritable in humans? And to study this, we'll be leveraging the Human Connectome Project dataset, which collected neuroimaging data from identical twins, fraternal twins, and siblings. So I'm sure many of you are already aware of connectomes or networks, or sometimes also called graphs. But to give a brief overview, connectome consists of vertices and edges A vertex here represents a region of interest or a region of the brain, and the edges represent some connectivity measures between a pair of vertices. And here we'll be leveraging the diffusion MRI, from which we can estimate the number of neuronal fibers that go from one region to another region. So to study heritability, we'll be leveraging statistical modeling for connectomes. The idea is that we assume that the edges that we observe in a connectome are random. And we can model this randomness via some distribution. And here we'll be leveraging distribution that are specific for networks. And once we have chosen a model or a distribution, we can estimate the parameters given our data. And once we have the parameters, we can use them for subsequent inference tasks. And here we'll be using the parameters to estimate our notion of heritability, which is slightly different than the traditional notions. So to measure heritability, it's a pretty simple process. We start out with a pair of connectomes, and we use what's called the adjacency spectral embedding. You can just think of this as an algorithm to estimate the parameters of our model or our distribution. And once we have this pair of two sets of parameters, we can compute the distance or the test statistics between the pair of parameters. So here, the distance is a measure of dissimilarity, meaning that the smaller the number or the smaller the test statistic, the more similar or closely related they are, and higher the number, the more dissimilar they are. We can repeat this procedure for each pair of subjects. So we can do this for each pair of identical twins, fraternal twins, and siblings. And as a control, we've also included pairs of unrelated individuals, which are defined as people who don't share either a father or a mother. So once we do this for each pair, we'll obtain a distribution of distances. This is the main result, and these are the distributions of the distances. So each row represents a familial type. Monozygotics are identical twins, dizygotics are fraternal twins, siblings, and unrelated. Each column represents a different measure of distance. But the idea is that, or the overall result here is that the distribution of the distances for the identical twins are shifted closer to zero. And we see this stochastic ordering from identical twins, fraternal twins, and siblings. That is, as we go down in more dissimilarity in genetics, we see a shift towards greater distances. So to explain each column a little bit more, on the left we have what I call exact, and this just measures if there are any differences between the two sets of parameters. And in the middle we have a global scale which measures, we say that we factor out some global constant from both sets of our parameters and then compute our distance. And on the right column, which I call vertex-wise scale, meaning we factor out some vertex-specific constants and then compute the distances. And what we essentially see is that even after factoring out the commonalities, we still see this ordering of the distances. And yeah, I guess for discussions, I can go more into detail about specific slides. If there were any questions.
Okay, thanks, Jung-Won, for the great talk on the on the human connectome heritability. Okay, I personally think it's a really interesting topic. And since we don't have questions from Q&A, I want to kick out the Q&A with one question from me, and we have plenty time to discuss. So one thing is, so this is all structural connectome, right? You probably collect the data yourself or from collaborator from the using diffusion MRI. I'm wondering, do you assemble voxels a little bit to get a more sparse connectome, or you actually the connectome is actually down to a very voxel level. I see you have voxel-wise. I'm just curious.
Yeah. So the vertex Y. So the answer to that is I use a parcellation, which divides the human brain into 115 different regions. So this is not at the voxel level; it's much more macroscopic. So we're talking about large regions of the human brain. But there exist many different parcellations of the human brain. I chose one. It's called AAL, but I have repeated this analysis on different parcellations, not at voxel level, but at parcellations up to 1,000 different vertices, and the answer basically remains the same. That regardless of your choice of parcellation, that you see this ordering of the distances.
I see. So my understanding about parcellation is basically RIs, like you kind of aggregate information within one RI to be one vertex.
Right, exactly.
Basically, it's more like a higher level of structure rather than down to a very neuron level comparison.
Right, and part of the issue here is that with diffusion MRI, you can't get neuronal level information. You can only look at aggregates because the resolution just isn't there.
Yeah,
so it's
I
certainly
understand.
different than say looking at electron microscopy or things like that.
I see. Yeah, but even like aggregation level, I mean, voxel is not just single neuron. It's like it's a group neuron that within a particle 3D in a 3D area, right? So that's still a kind of aggregation, but still you can further zoom out to be parcellations or to be RIs that you further aggregate this information to represent the non-neural activity, the like a vertex for a specific structure. So yeah, that's good to know just at which level the information is lays on, because it's possible that when you get to a very lower level, you don't see very strong similarity or a very close distance across people who don't have any relationship. But I think it's more interesting that when you zoom out to a higher level, you still see a large distinction between twins and between the people who don't have relations. So that's more convincing rather than when you get down to a very neuronal or voxel level of comparison. That's my personal idea. I'm not sure that that's even valid.
Yeah, well, I mean, the idea of I personally use parcellations because many neuroimaging studies like that uses connectomes will typically use parcellations rather than voxel-wise connectomes. I mean, I can certainly redo this analysis in voxel-wise space. That's certainly not a problem. But it just wasn't a question that came up, but certainly an interesting one and definitely something that I can look into.
I see. Great. Thanks for the answer. Oh, question from Patrick?
Yeah. Thank you for your talk, first of all. This is really interesting. The one thing I was interested in was like, how does age play a factor into these results? Like, are you seeing as like a subject's like age progressing? Does the structural connectomes, like, does that increase in distance?
Right. So the one analysis that I've done is with, well, it's a little bit weird with identical twins because, well, they're going to be the same age. But what we have done is looked at the relationship, the correlation between the difference in age, say in siblings, and the distance themselves. So what we essentially find is that there's no relationship between age and the distances themselves. But I mean, I'm sure there are, I mean, I know there are literature that says that, age and variety of other factors affect connectivity. It could potentially be just because of the pre-processing pipeline that I used that didn't show up. I mean, there are many reasons why that could be. So But in my analysis, that there was basically no correlation between age and distances.
Cool, thank you.
Yeah, I know that age is a big factor for analyzing functional MRI, but I'm actually not quite sure for just structural MRI whether the age can alter so much things. Like for the structure.
I guess we can easily just kind of look at this and say that, I mean, I guess for the most part, the subjects in this particular study are within very similar age range, and there's very little variance in terms of that of the identical twins, fraternal twins, and siblings. So one can assume that maybe because they're in the same age range that I didn't see any age effect.
Cool. Thanks, thanks, John 1 again for giving such a great talk and also answering all these questions. Very interesting. So now let's move on to the third talk, which is also the last talk for this session and probably the whole conference. Let's welcome Patrick Zhang, who is a researcher volunteer from Azadeh Yazdan's lab from University of Washington. He's going to talk about does functional interconnectivity reflect an anatomical connectivity, a simulation study. Okay, so Patrick, just take it over.
Okay, cool. Let me present my screen here. Okay, awesome. Yeah, thank you for the introduction. My name is Patrick. I'm a research volunteer at the Yazdan lab at the University of Washington. And this is actually my first talk at any science conference, so thank you for giving me the opportunity. And today I'll be talking about whether functional connectivity reflects anatomical connectivity and how we used a network simulation to study this relationship. Sorry. So first, what is anatomical connectivity and why do we care? Well, if we're interested in the connectivity between two regions of the brain, A and B, one way to define this connectivity is by looking at the synapses from neurons in region A to neurons in region B. This is called anatomical connectivity, and it's probably pretty challenging to calculate in an experimental setting. So researchers often are interested in functional connectivity as well, which tries to measure the strength and relationships of dynamics between the two regions. Functional connectivity can be derived from some kind of brain signal, such as local field potential or LFP. And there's multiple types of metrics we can use for functional connectivity. And so in our study, we're interested in how each of these metrics reflect the underlying anatomical connectivity of the brain. So we're using a network simulation to study this, specifically a network model of a cortical chunk. And because it's the simulation, we have full visibility into both the positioning of neurons and synaptic weights between neurons. And so we can use this information to derive anatomical connectivity. And we also wanted to record local field potential from our simulation and use this LFP to calculate different functional connectivity metrics and then relate that back to anatomical connectivity. So our model is 1.5 by 1.5 by 2.6 millimeters in geometry. We're simulating on the order of 100,000 neurons, and we're recording LFP from 54 recording sites laid out as 3x3 grids on the right. And we're simulating for 40 seconds and using a tool called Vertex for both the modeling framework and the initial model parameters. And we're running our simulation on the Neuroscience Gateway portal. So once we had our model, we wanted to validate that the behavior of the model was relatively biophysically realistic. So I'll briefly go over some of the checks that we did. So first is we looked at a spike raster plot of all of the neurons in the model, grouped by their cell type and their positioning within cortical layers. So the horizontal change in colors in this figure represents a different neuron group, totaling 15 different neuron groups. Here we just wanted to check that the firing dynamics per group were different. And then for a single neuron group, we wanted to check that the firing rate histogram was a relatively log normal distribution. And then for a single neuron, we wanted to check its interspike interval histogram. And here we were looking for a Poisson distribution. Because our model is generating LFP, we also wanted to verify that the LFP generated was realistic as well. So here we're looking at a power spectral density curve of an LFP recording from the center electrode on the surface of the model. And we're looking for a 1 over frequency curve here. You can also notice that there's a bump around the gamma frequency at around 40 hertz. This is an artifact from our initial model configurations, and we're actively looking at how tweaking these configurations can change the size of this bump. So next, we computed a few connectivity metrics from our model. So for functional connectivity, we looked at coherence, which is a pretty common metric in neuroscience. Pairwise Granger causality, because that introduces a metric with directionality. So A's connection to B might not equal B's connection to A. And then partial Granger causality, because that introduces conditionality as well. So if we're interested in A's connection to B, it tries to account as much as possible for any other node C that could potentially be contributing to A and B's connectivity. And then for anatomical connectivity, the way we calculated this was by looking at all the synaptic weights in the model, inversely weighted by the distance from respective neurons to the recording sites. So I wanted to share some really preliminary results with the disclaimer that there's a lot more analysis to be done on both the model side and the connectivity metrics side. But one of the things we were interested in is the correlation between each of these functional connectivity metrics to underlying anatomical connectivity. And we're looking at this per frequency. So per frequency value, we have a correlation coefficient from each of the connectivity metrics to anatomical connectivity. And this correlation is calculated across recording sites, pairs of recording sites in the model. So a few interesting things we noticed. Partial Granger causality, the red line in the graph, seems to be performing the most consistently across frequencies. Both of the Granger causalities seem to be outperforming coherence at higher frequencies, specifically higher than 150 hertz. And there seemed to be a dip in all three of the correlations at around 40 hertz. If you recall, this is also the frequency range where we saw a bump in LFP. And so our current thinking is that whatever mechanisms in the model are causing that bump in LFP could potentially also be contributing to this dip in correlation. So for next steps, we want to investigate whether tweaking the model parameters affects the relationship between functional and anatomical connectivity. Specifically, if we tweak the parameters to get rid of that bump in LFP that we saw, does that also change the correlation at around 40 hertz for our metrics? There are additional functional connectivity metrics that we're interested in and other ways of calculating anatomical connectivity, such as only looking at excitatory or only looking at inhibitory neurons. In the longer term, we want to extend the model to simulate stimulation at certain sites, and then also study effects of potentially adding a plasticity rule such as STDP in the model. So I wanted to thank everybody at the Yazdan lab for giving me an opportunity to work on such a cool project. Professor Yazdan and Professor Shea Brown for their guidance, and my co-author Julian for all of his mentorship. Thank you very much.
Okay, let's thank Patrick for giving such a great talk. I think it's very interesting. But it's probably a short talk, so there are a lot of technical details that are hidden from the slides that I'd like to know more about. I guess people from the audience might have similar interest. Since right now we don't have a Q&A, let's just kick out with a question that I have. I'm just curious, can you briefly talk about what kind of model you build to do the simulation? Like, do you have that kind of info? Like, what kind of model you build? How you like basically. My understanding is when you have a neural network in theoretical neuroscience, you kind of have a bunch of connections and which are represented in W. But these W are kind of a given weights, especially when you try to do simulation. And given the structure you build, how you got the functional information that is different from the structure connection that you build, because usually when the weight is strong, then presumably your functional signal should be strong. That is very correlated with your anatomic connections. How is that true, or it's actually different?
Yeah. So a few things about the model, like the one unique thing. Well, so we're using a modeling framework called Vertex to do all the modeling. The one unique thing about that is it's able to generate LFP from the model, specific to like the questions about like the characteristics of the model. So basically you set up the types of neurons that you want in this model, and like where they are lying, like where their positioning is in terms of the cortical layer. And then connections across neurons are calculated. Let's see, I have a slide here. They're calculating by using a 2D Gaussian spatial profile to model decay of connection probability. And they're also calculated like per groups of neurons in certain groups to connected to other neurons or certain groups are also have different like parameters for this Gaussian spatial profile. So yeah, that's, I don't know, does that answer your question?
Yeah, I think, yeah, that's kind of more complicated than I expect with this tool, I guess. And just another minor thing, and I'm curious, do they also have the kind of the mechanism with like the ramping up, like the potential spike and all this kind of neural mechanism existing in single neurons that really mimic the real physics process and triggering this LFP and spike trains?
Yeah, so the spiking model they use is an adaptive exponential integrate and fire model. So my understanding there is like it's an integrate and fire model, but it like because it's adaptive exponential, it's able to generate a lot more different firing patterns. And then like the activity is driven by like random currents into each of the neurons, and that's modeled as a Ornstein-Uhlenbeck process. And so we, as the users, we configure the mean and standard deviation of that process.
I see. Cool. Thanks for that answer. One question from the audience: was the model on a certain area of the cortex or brain, or the entire brain?
So our model geometry is pretty limited. We didn't specify a certain area of the brain. I think it's just pretty generically a cortex. But the neuronal types were borrowed from the original Vertex tool.
I see. Thanks. Hope that answered the question from Catherine. Yeah, we still have maybe time for one or two more discussion points. Maybe, yeah, I see Patrick list a bunch of discussion points he want to discuss. I'm also curious to learn more about that. Yeah, maybe the first question, what are some other ways to analyze the anatomic functional connectivity relationship? Here you show three metrics, I guess, or three ways: the granular causality, the pairwise and stuff, and also the correlation.
Yeah, so I was actually hoping on getting some feedback from the group here. Like right now, we're just doing a very simple correlation between some of these metrics, but I'm interested in like if there are like other ways of doing this, and if there are other like interesting like ways of calculating anatomical connectivity that could be useful for this analysis.
Yeah, okay. That's a bit beyond my knowledge. I'm not sure I can give you some expertise, like feedback on that. But yeah, for people who might know more about this, welcome to talk to Patrick offline to provide him feedback and some further discussion. Yeah, I think maybe one more time, time for one more topic to discuss. Maybe go to yeah, good. Yeah, cool. I'm not sure that there is very certain thing you want to mention, but one thing actually confuses me a little bit when I see the analysis is the representation of the functional connectivity. Like I know for anatomic connectivity, you have to weight, which are the parameters or the numbers you can try to analyze with. For the functional thing, is that just LFP? Do you kind of, it's actually not very clear that how you apply this granular causality on LFP signal only. I mean, for anatomic connectivity, you know the structure, you know the weight, you know the edge. Maybe it's more straightforward, but for LFP-based functional connectivity, I'm not quite clear how to.
Yeah, so the all the functional connectivity metrics are calculated from the LFP measured in the model. Specifically how they're calculated, full disclaimer, my co-author Jillian did the actual calculations, but for like something like Granger causality, what it tries to do is it will try to, like, at any given point, it looks at how past data from one of the LFP recordings can predict current data for the recording you're interested in. And so using kind of that method, it derives like some connectivity value for a pair of recordings.
I see. Okay, that makes sense actually. Basically, try to use the. I see. Just use the LFP from one neuron, try to predict the other thing. The other neuron, try to see whether they have a strong functional correlation from LFP or not.
I guess one thing to clarify is like the LFP isn't coming from specific neurons; it's coming from, I guess, like a population of neurons in both in the model and during experimental settings. So
would that be more clear or more specific to individual neuron if you use spike trains instead of LFP?
Yeah, potentially. I think in our study we were kind of interested in like how like metrics from LFP would reflect back to the anatomical connectivity, because LFP seems to be a pretty common way of recording activity in the brain. And so we wanted to see that like the connectivity results coming from LFP actually had some base in anatomical connectivity.
I see. Okay. Yeah, I think LFP might be more like related to the anatomical structure across neurons in a population rather than single neurons spectrum data. That makes more sense. Okay, thanks for the answer. Yeah, I think that's basically. Yeah, we're running to the end of the session given time. Let's give applaud to the 3 speakers today for the session: Rusbek, John Won, and Patrick, for the great talks on these study on different morphology, structures, connectome, connectivity. And I think this kind of topic is really interesting. Like it combines knowledge from graph, from statistical modeling for connectome, all the stat, to very neuroscience stuff, which is a really cool interdisciplinary research area that I'm personally very fond of and excited about. So thank you all for giving such a great talks, and thank for the participant from the audience. That's basically the end of session. And one thing I want to mention is there is one more session, which is the NeuroMatch 3.0 closing session on stage by Conrad Corden, coming in maybe 10 minutes. So people are welcome to join that last session. And to basically try to have fun celebrating together for the end of this big conference contributed by so many people's great work. Yeah, that's basically it. And nice to know you guys and to be able to host this whole session. I hope to see the great cool research in the future. Okay. Okay, cool. See you guys and have a good day.
Thank you. Bye-bye.
Bye.